Good evening, all students and respected parents. Today, NEET 2026 re-examination just now completed. Just a few minutes back, the students have completed. So they have faced this challenge another time in giving this Re-NEET examination 2026. As everybody knows, May 3rd, first the exam was conducted, and then there was certain circumstances because of which it had to conduct again by NTA, right? But whatever it is, let us not go back or dig back what happened earlier. Now let's stick on to how the Re-NEET examination today, how it has happened June 21st, 2026, and how is the paper subject wise: botany, zoology, physics, and chemistry. And our expert faculties are working on preparing the solutions for all the questions to explain to you, right? And also, there is a team of faculties who have reached the examination centers, and they are speaking to our resonant students and other students also to understand, to know what are the challenges and how the paper in comparison to previous year papers and all, right? So the most common, the most common feedback from majority of the students when it was spoken at the examination centers, what we got is in comparison to May 3rd NEET 26 exam, May 3rd exam, this is a NEET examination. Coming to biology, it is little more comfortable in comparison to May exam, right? That means the students are able to more comfortably solve more number of questions to see in biology part. So that's a good news like for the students, because of 50% of the paper will be having the weightage of biology only, botany and zoology together. May not be exactly 45 will come, but closer to both together 90 questions will come, right? And even the chemistry paper also, little more comfortable in comparison to May exam. May 3rd exam happened. So what does it mean? That the student who was solving, suppose let us assume out of 45 questions, he was able to solve comfortably 30 questions. Now he is able to little more. 30 questions he is doing anyway, and he is comfortable in solving these 30 questions and also solving little more number of questions. Right. So this is the first feedback what we got for biology and chemistry. Coming to physics, it is opposite to biology and chemistry, but still it may be like a few of the students we have spoken that this is the general pulse, but when the paper is obtained, right, different students will be feeling in a different ways, because the way you have given the weightage, the different types of chapters you will be liking. Some chapters are very difficult for you but very easy for other student, and vice versa also. So in that case, as a general opinion, what we have observed in physics, little opposite happened in comparison to biology and chemistry, that is a little more challenging in this June 21st exam in comparison to May 3rd exam, right? But having the physics difficulty level little increase is not a concern, should not be considered as a concern, because it is not individually for you, it is for everyone. So overall effect will be there, that any student, none of the student need not worry about that. So biology comfortable, chemistry also comfortable. Coming to physics, little more challenging than previous. And coming to biology, there are few of the chapters I have collected and I will share with you. There are some chapters in biology where more frequent, repeated number of questions came, more weightage of questions came, are from sexual reproduction in flowering plants, human reproduction and human health, evolution, ecology, biotechnology, genetics, etc. So these are few of the chapters where more number of questions were seen. More number of questions were seen. And coming to chemistry, the questions were seen from across the chapters. Like you can see the questions from starting from the periodic table and chemical bonding, and then P block general properties questions are observed. Even in D block, multiple questions I have seen. Coordination compounds also multiple questions I have seen. From last year 2025 NEET exam paper onwards, I am warning you, I am telling you, I am alerting you that D and F block plus coordination compounds is a very, very important set of chapters. Even you are studying up to NCERT level also, then also you will be getting easily 4 to 5 questions out of 45 questions. D and F block plus coordination compounds together. Weightage wise, how you have to share it, how you have to practice it, that all we will be coming back to it. For the entire chemistry subject, I will be explaining. Even for biology and physics also, I will welcome the expert faculties, so that those students who are preparing for the next year exam, for them it will be working as a guidance where to focus, how much to focus, right? And what is the weightage of different chapters, right? From hydrocarbons questions were seen, chemical kinetics, electrochemistry, coordination compounds, then chemical bonding, then chemical equilibrium and ionic equilibrium, like that. Different from different chapters questions came even in chemistry also. And from physics, little more feedback I need to collect here, but the roughly what I have listened to the expert faculties is the number of questions from 12th portion is a little more than of the 11th portion. So keep this in mind. Not only in a NEET, even in the other competitive exams like JEE also, 12th portion weightage will be higher in comparison to 11th portion. Like 12th portion will be like 55 to 60%. So if 12th itself is taking 55 to 60%, what about 11th standard? It will be from 40 to 45%. So this variation 5 to 10% extra will be available, will be coming from the 12th standard, because obviously you also can understand not many chapters are there in 12th standard, but 11th chapters are also important. How they are important is, these all the 11th chapters, their continuation whatever is there will be having a strong grip for the 12th chapters, right? So the foundation of 11th standard will be very important for 12th standard. Like as an example, if you see in chemistry, uh, general organic chemistry, IUPAC nomenclature, structural isomerism, hydrocarbons, like different techniques in physical separation of organic compounds. All these are studied in 11th standard, which will be very much needed. GOC, you can, uh, resonance, mesomeric effect, inductive effect, then a hyperconjugation, acidic strength, basic strength, tautomerism, carbocation rearrangement, then carbanion stability. All these things are very, very needed in 12th standard, different reactions and their mechanisms. Yes or no? So in this manner, there will be a link between 11th and 12th standard chapters. That is the reason why 11th chapters are very, very important, and they make the 12th syllabus, the 12th understanding to very, very strong and high. Got the point? So in this manner, you have to prepare. And now coming to overall chemistry part. We know that chemistry part is in the three parts, right? Organic, inorganic, and physical chemistry. Coming to inorganic chemistry, very, very easy part it is, that maybe little weightage is lesser, obviously. Like if organic and physical are taking 35%, inorganic may come to 30%, or even little lesser also, 25% also. The highest number of questions will come even by the level wise also from the organic chemistry greater than physical chemistry, greater than inorganic chemistry. Every student have to keep this point in mind. and accordingly only you have to put your efforts also in the preparation, right? So before we get the questions by the expert faculty to discuss with you, right? Our test paper discussion, the sequence will be like first botany will be there, then zoology, then physics, and then chemistry. And all the faculties are making the PPTs for you to explain the questions in a very beautiful manner. And whatever the tips and tricks are there, or different methods are there to solve the questions to reach to the accurate answer, correct answer, they will be teaching to you. So very knowledgeable session it is. That stay tuned and be till the end of the video, entire paper discussion. And also at the end of the test paper discussion, there will be test paper analysis. The entire paper will be analyzed by the subject experts, like in physics, the paper analysis, chemistry, botany, zoology, etc. Right? That also will be very, very helpful for you in the upcoming exam preparation. There may be some students who will be giving a second time, they repeat the exam, they give the exam again, or even multiple times also they give. So for such type of students also, this resonance junior colleges or whatever live paper discussion solutions are there, they will be very, very helpful to you. And also at the middle of the paper discussion, we will be releasing the key sheet, right? For a particular code, we will be announcing the code also, test paper and key, both we will be releasing it. And if anybody is there who want to have this test paper and key, in the chat box, you can keep your number. So we will send it through WhatsApp to your number, whatever the test paper and key is there, right? And coming back now, let's focus on a very important point, that is, in chemistry, whatever three parts are there, as just before I was telling you, inorganic, organic, and physical chemistry. Inorganic chemistry, number of chapters are limited. Suppose if you see periodic table, chemical bonding, P block, physical properties, then D and F block, coordination compounds. Right. So these are only the chapters. Na? So these chapters, number of chapters are very less, and weightage is also good, because they are the chapters even you are sticking to the NCRT, not 100,000% sufficient. And if you take all these 5 chapters, then the weightage of the number of questions out of 45 questions, you will be getting around 10 questions. 10 questions from very easy theoretical chapters, just reading NCRT is sufficient. So you understand it, how much it will be helpful. 10 questions means straight away 10 into 10 to 12 questions, right? We cannot exactly say, maybe 1 or 2 questions lesser also may come in some sessions, like it will come to 8 to 9 questions also. On an average, if you take even 10 questions also, 10 into 4, 40 marks you will be getting from inorganic chemistry alone. And believe me, if you are preparing through NCRT, definitely it is possible to score 40 out of 40. This is one part to prepare and keep it in your pocket, right? The second part is physical chemistry. What chapters are there in physical chemistry? Starting from mole concept, atomic structure, chemical and ionic equilibrium, thermodynamics, electrochemistry, chemical kinetics, then solutions and colligative properties. So these are the chapters which are there. Have I missed any of the chapter? No, na? These are the chapters, 7, 8 chapters, whatever are there. These are the chapters which are having, even you are sticking to NCRT for the content wise, but a little calculation wise, the formula wise, little application part to some moderate level application is needed in this physical chemistry. But the beauty is that whatever the content is there, concept is there to the NCRT, that much is sufficient. You need not go with new formulas, new equations and all. Content wise, NCRT is sufficient, but in a different perspective, in a different angles, the questions can be asked, the data can be given to you, right? So This much if you are focusing in physical chemistry, that also will be helping you in getting a good number of questions. Maybe majority like 13 questions we feel on an average 13 questions coming, 13 to 15 let us assume. But even you take a 15 also, 15 into 4, 60 marks it is. 60 marks it is. Right now. So that is another most important factor. And within this one also, you can select among yourself that you can select within these chapters that which are the chapters which I want to complete first. Like atomic structure is there. Atomic structure is such a chapter which you will be getting majority of the portion even in physics also, right? In modern physics, atomic physics and nuclear physics, etc. Bohr's model will come, dual nature of atom will come, then spectrum will come, like different concepts will be coming. So as this chapter will be coming again in physics also, so 2 times reading will be helpful for both the subjects. They don't see that atomic structure Bohr's model they have given in chemistry, so they should not give in physics. They will be, it is independent that whether it is in from that particular concept, the question is given in chemistry or not, they will be asking in physics, vice versa also. Understanding? So this is another first important point. So you can select atomic structure. Second, mole concept. Without mole concept preparation in physics, physical chemistry, it will be very difficult for other physical chemistry chapters. Therefore, preparing for mole concept is always advisable. Very strongly it is advisable to prepare for physical chemistry, mole concept chapter. That is stoichiometry only, right now. So, and linked with the mole concept, there is another part is there, that is redox reactions, where oxidation, reduction, redox reaction, balancing of redox reaction, types of redox reactions, equivalent concept to some extent we will be studying, right? So this is also very important as this is the second part of mole concept. So this mole concept plus redox reactions together is one set. It is mandatory for everyone. It is mandatory for everyone. Right. Then coming to third, thermodynamics and thermochemistry. Thermodynamics part up to first law, even it comes in physics also. Therefore, you can take the support. The time will be saved as it is there in both the subjects. Thermochemistry is the part which you need to put the additional effort. Right. But that is comfortable only. Every student can do it. And then chemical and ionic equilibrium is one logically some individual chapter, you can say, not completely individual, but majority concepts are new in this one. So that is your wish, like you want to prepare for chemical equilibrium, ionic equilibrium, different concepts will be there under this also. So that is up to you, right? Then coming to 12th standard physical chemistry, colligative properties is there. Right, concentration terms and colligative properties. Concentration terms is something like a revision to the mole concept stoichiometry you study in 11th standard. So that part will be anyway covered. And coming to second part, colligative properties, 4 colligative properties: elevation in boiling point, depression in freezing point, osmotic pressure, and relative lowering of vapor pressure. And Van der factor, anyway it is there for dissociation association, right now. So this is a very easy chapter. Very limited number of formulas are there, which by easy calculation itself we will be getting the answers also in this chapter. So solutions and colligative properties is also one advisable chapter within physical chemistry. And next chapter is chemical kinetics, then electrochemistry. These two chapters, literally to say, these two chapters are challenging chapters only. So it is all up to the student. Suppose if you are comfortable in all these chapters, right, so put some extra efforts so that you can prepare for the chemical kinetics first, and then electrochemistry also later on, because from chemical equilibrium, electrochemistry, chemical kinetics, thermodynamics, interlinked concepts also are there. Interlinked concepts are also are there. So majorly in the competitive exams, they will be focusing on this type of questions also, where from the two chapters knowledge is required. Right now, so you have to list down all that concepts which are having interlinking between the chapters. Right now, so these all about physical chemistry, how to handle. But physical chemistry weightage is more than inorganic chemistry. Understand it. Then coming to organic chemistry. Within organic chemistry, the whatever the intro part is there, that is means fundamental concepts whatever are there, they are all kept in the 11th standard syllabus. Like to start with IUPAC nomenclature and structural isomerism, then to some extent stereoisomerism is also there, like a geometrical, optical, a little will come, conformational isomers and all. Then comes to general organic chemistry part 1 part 2, where we will be studying inductive effect, mesomeric effect, resonance, hyperconjugation, aromaticity, electromeric effect, then acidic strength, basic strength, tautomerism. All these concepts will come. Carbocation, carbanion, free radical, carbene intermediate, benzyne intermediate, etc. Right now, so these all concepts are very important when we are going to the 12th standard. As a logical point, I will tell you, understand it. Altogether, let us assume from the 45 questions, if we are getting 15 questions from organic chemistry, roughly let us assume 15 questions from organic chemistry, 5 to 6 questions will be coming from 11th standard syllabus, because there is no much high concepts, very limited concept is there in 11th standard organic chemistry. But 12th standard chemistry is majoritarily organic only. Majoritarily 12th standard chemistry is organic chemistry only. Lot of reaction mechanisms are there, named reactions are there, rearrangements are there, exceptions are there, different applications are there, interconversions are there. So different concepts are there, so many concepts are there, that obviously the weightage of the organic chemistry is very high in 12th standard. That is one of the major reason that students will be feeling very challenging when they are solving for solving the questions in organic chemistry. But this is the zone where students have to strongly focus into and make the organic chemistry portion comfortable. As already I told you, again I am telling you, the number of questions from organic chemistry will be more than the questions from physical chemistry, than the questions from inorganic chemistry. This is the order of weightage of number of questions. So accordingly, obviously, our preparation also should be there. Yes or no? So this is the overall way how to solve, how to balance the time between the different types of chapters within chemistry also. Understanding, enough? And coming to whatever another most important task is there within a NEET examination, that is time management, right? So for students who have already given the NEET exam previously, 2025 or 24, or even May 3rd examination, whoever has given, you will be having the idea. But think of yourself, was I able to manage the time properly? Because 3 hours is the time duration given to the students. Within this 3 hours, 3 hours means what? 180 minutes. 180 minutes. And this is the 180 minutes given to solve 180 questions. That means 1:1 ratio. 1:1 ratio. So, every question from NEET examination should be completed in 1 minute. But how is it possible for physics and even chemistry, where calculations are heavy? So in these subjects, the time management, the time balancing will be with the help of botany and zoology, right? So whatever the 180 minutes is there, ideally what we advise to the students to prepare very strongly with the multiple times of revision in botany and zoology, that the entire biology will be completed in 40 to 45 minutes. 40 to 45 minutes. So if 40 minutes of biology is completing, the another 140 minutes will be left over for physics and chemistry. And within this 140 minutes in physics and chemistry, chemistry is the second easier part, and third will come obviously for the NEET students. Third we will be keeping the physics part. So you need to take care in such a manner that the time division management should be in such a manner that minimum to minimum you have to give 70 to 75 minutes for physics, even 80 minutes also for some of the students who do the calculations little slower or feel the challenge in questions, right? So if you want to get 70 to 80 minutes for physics, definitely you should complete chemistry within 60 minutes. So first 40 minutes biology, 60 minutes chemistry, then 70 to 80 minutes from the for physics part. So in this manner, if you are putting the weightage in solving the questions, it becomes very very comfortable for you in reaching, in touching, in solving maximum number of questions. The game in a NEET examination is solving more number of questions. Quantity matters in NEET and JEE main examinations, but in JEE advanced examination, quality matters. Selective number of few questions are done, that is okay. But in a NEET examination, more number of questions should be solved so that you will be getting more marks. By getting these more marks only, the seat allotment confirmation will be possible. Right now, like in the Telangana state, if you see 480, 485, the last cutoff marks, right? Let us assume approximately 500 to the safer side, 10, 15 marks extra. So 500 marks, and for the Andhra students, if you see, it will be beyond that, 550, 560, 580 they have to get according to the college. And different states are having different level of competition, that the local whatever government colleges are there within that state, for them the number of marks required will be varying from state to state, right? For the Telangana students, luckily it is like comfortable only, but other states tight competition will be there in this number of questions, number of marks to get the MBBS seat, right? So now let's start with a botany paper discussion. I welcome our Amulya ma'am to continue with the botany questions discussion. Please, ma'am.
Thank you, sir. So, very good evening, dear students. So, as you have just completed your Re-NEET examination, and as we have just gone through the biology paper, the first, the botany questions, like the botany questions, what I felt is they were straight away from the NCERT, and these questions were very easy, right? And the students who have gone through the NCERT thoroughly, I believe that you have given your best, right? So with this, let us hope that your like the scoring and your marks and everything, and may God give you what do you say, the successful life in this, the NEET journey and everything. And let us solve the questions of the botany questions now. So let's see here the first question, right? How many turns of Calvin cycle are required for the formation of 3 molecules of glucose? So as you can see here already, the name of the chapter has been mentioned. It is from the respiration in plants. So you can see here how many Calvin cycles are required. Such an easy question. We know that to form, what is that? The glucose, isn't it? I mean, right, sorry, the chapter name, you can just check it out here. So it is see here how many turns of Calvin cycle are required for the formation of 3 molecules of glucose. So as we can see here, my dear students, 3 molecules of glucose. For 1 molecule of glucose, for 1 molecule of glucose, we require, right, 6 turns of Calvin cycle. 6 turns, isn't it? For 1 molecule, to form 1 molecule of glucose, you require the 6 turns of the Calvin cycle. And what he is asking how many cycles do we require for the 3 molecules of glucose? So it is the photosynthesis, है ना? This is from the photosynthesis wala. Right. So here you can see for 1 glucose, if it is 6, so for 3 into 6 you will get 18, isn't it? So our answer is the first one. The first one. Oh, right. Let's go to the second one. Right. In photorespiration reaction, right, catalyzed by Rubisco is shown below. So this is from the respiration in plants. The previous one was from the photosynthesis and glucose formation and all that. So here you can see photorespiration reaction catalyzed by Rubisco is shown below, right? Photorespiration, we know this is one of the reaction that takes place in a C3 plants, isn't it? A process which is basically of no use. It is, you can see, one of the waste process in the C3 plants, isn't it? Wherein the Rubisco, we know the Rubisco has got a dual nature. It also acts as a carboxylase and it also acts as a, what is it, oxygenase, isn't it? Right? So here the Rubisco, as it acts as a oxygenase enzyme, है ना, right? So when it RUBP binds with the oxygen, it forms 3-phosphoglycerate, that is one molecule of 3-phosphoglycerate. And also what is the other one? The other one is the 2-phosphoglycolate, isn't it? It forms, that is 3-phosphoglycerate, and it also forms a one molecule of the 2-phosphoglycolate. Straight away question from the concept, that is photorespiration. Let's move on to the next one. This is from the plant kingdom, and the concept, that is the gymnosperms, isn't it? Right here it says the statement 1. In gymnosperms, the male and the female gametophytes remain within the sporangia, isn't it? The male gametes, right? In gymnosperms, the male gametes remain in microsporangium, right, microsporangium, and the female gametes are formed within the megasporangium. So the statement 1 is absolutely correct, right? Let's move on to the next one, statement 2. It says that in gymnosperms, seeds are not covered. Yes, absolutely. Gymno, and naked seeds. The seeds are not covered. So both the statement 1 and the statement 2 are correct. This is from straight away the gymnosperms concept from the plant kingdom. So both statement 1 and statement 2 are correct. Okay, let's move on to the next question. Now, how many molecules of pyruvic acid are produced? Now he's asking about how many molecules of pyruvic acids are produced at the end of glycolysis from 206 molecules of glucose. Very easy. We know, my dear students, in a glycolysis, है ना, what happens, right? One molecule of glucose, isn't it? One molecule of glucose, right? Which is C6H12 and O6, isn't it? One molecule of glucose during the glycolysis, wherein the partial oxidation takes place, this splits or this breakdowns into two molecule of pyruvic acid, right? It forms two pyruvates or two molecules of pyruvic acid. So he says now, if there are 206 molecules of glucose, how many molecules of pyruvic acids are formed? If from one glucose two pyruvic acids are formed, therefore from 206 how many are formed? So this is 412, right? This is 412. Obviously, 206 into 2, right? So our answer is the first one, that is 412, right? Let's move on to the next one. See, most of the questions which I'm here to discuss, all the 15 questions or 16 questions which I'll be discussing here are from 11th standard, or straight away from the concept, straight away from the NCERT. Let's move on to the next question. This is from, the biological, right, classification, and here you can see the match the following. This has been asked from, that you have kingdom mycota, isn't it, and wherein, right, sexual reproduction ka concept, isn't it? They undergo sexual reproduction and also asexual, they form spores, vegetative fragmentation, right? So let's see what he is asking. He says that match the correct option. Fusion of protoplasm. So protoplasms are fusing, so it is nothing but straight away, that is plasmogamy. Protoplasms, fusion of protoplasm is nothing but the plasmogamy. So this is the second one, right? Fusion of two nuclei, nucleus of the two gametes, they are fusing here. So what is happening here? Karyogamy. Karyo ka matlab is nucleus. So here you can see this is karyogamy, right? And then generation of haploid. So the cells produce the diploid cells, they again produces the haploid cells by undergoing meiosis. So meiosis between the gametes. So very easy, right? Fusion of protoplasm, that is plasmogamy. So A is 2. Let's go with this. A is 2. Okay. Fusion of two nuclei. Nuclei ka matlab karyogamy. So B is 3. So B is 3 here, right? Generation of haploid, meiosis between gametes. Okay, right? So this is C1. So option is second one, right? Let us move on to the next one. Mitochondrial inner membrane encloses. Very easy, right? Mitochondria. So this is from the chapter Cell, the Unit of Life, isn't it? And here you can see the mitochondria encloses, right? Obviously, it encloses the matrix, right? So such an easy question. Then phyllotaxy or phyllotaxy is the pattern of arrangement of. So this is from the chapter, right, morphology of the flowering plants or in the flowering plants. So you know, right, the phyllotaxy means leaves, the arrangement of the leaves, isn't it, is what we call it as phyllotaxy. Right? Arrangement, how the leaves are arranged. Are they arranged alternatively, right? Or they arranged oppositely, or they arranged wholly. That means right from each node a group of the leaves. Moving on to the next question. Mad cow disease is caused by. The mad cow disease, again this is from the chapter biological classification, the last few concepts where we study about the viruses, viroids as well as the prions. So the mad cow disease is caused by, we know it is caused by the prions, isn't it, due to the right abnormal foldings of the proteins, right? Moving on to the next one. Cell theory was formulated by such a easy question, right? This is from the chapter Cell, the Unit of Life, right? So cell theory, we all know it was given by Scleden and Swan, right? They proposed the cell theory, which was later then, right, the changes and modifications were done by the Rudolf Virchow, right, where he said the cells arise from the pre-existing cells, right? Which of the following plant growth regulators promotes internode elongation prior to flowering in cabbage, right? So the internode elongations prior to, right, flowering in a cabbage. So my dear students, what is the exact answer, right? Internode elongation. So this is nothing but, right, bolting. This is nothing but bolting. So he's asking about a bolting, the hormone, right, which is responsible for the bolting. And we know the gibberellic acids are responsible for the bolting and the malting process. So the answer will be the gibberellic acid, that is gibberellin, right? Okay, moving on to the next question. Which pigment has absorption peak at 700 nanometer in the photosynthetic reaction center PS1? Photosystem 1. So they have given us the 700 nanometer and they are asking us, right, which pigment observes this, right? We know there is a chief pigment which observes this, and the chief pigment is the chlorophyll A, whereas the chlorophyll B is the accessory pigment, right? And the other can also observe the wide range of the wavelength, isn't it? So our answer straight away goes with the chlorophyll A. Moving on to the next one, the plant kingdom chapter, right? Spinopsida, right? This is again, as I have told you, that most of the questions right here, right, which I am explaining you, all of them are from the 11th standard, and that too very easy. So it says that Spinopsida class belongs to, right? So we know, dear students, this is a class of the pteridophyta. Pteridophytes are further divided into 4 classes, isn't it? They are divided into Psilopsida, Lycopsida, Spinopsida and Pteropsida, right? So Spinopsida is one of the class of these pteridophytes. So we know there are 4 classes, right? Then further, moving on to the next question. Which of the following plant growth regulators is used as a herbicide, right? Again, this question is from the chapter Plant Growth and Development, right? So what's the question here? He is asking about which of the plant growth hormone is responsible for, right, or you can say which is used as a herbicide. What do you mean by herbicide? Right side ka matlab is killing, right, which kills the herbs, isn't it? Right, which stops the growth of these herbs, isn't it? Right, so we know that 2,4-D is used as a herbicide. And what is it? It belongs to the plant growth hormone. What's that? Auxin, This is a synthetic one, right? This is a synthetic auxin, right? And it acts as it, what is that? Herbicide used in the preparation of the lawns, vagera, right? So we'll check it out the other things also. Okay, done. Moving on to the next one from the chapter Living World. And the question is, genus represents, right? Such an easy question. You have those 7, isn't it? What is the taxonomic categories? The obligatory one, isn't it? And here it says, what is a genus? Genus is nothing but a group of related species. And now what is the species? A species which share the similar fundamental similarities and also are capable of interbreeding. So if you look at the option, it says that group is a closely related, families, no. Family is the next category, है ना, above the upper one. An individual plant or animal, no, right? A population of plants and animals, no, right? A group of closely related species, this is the correct one, right? Genus is a group of closely related species. Then moving on to the next question, the plastid that stores xanthophyll is, right? The plastid that stores xanthophyll. Okay, right. So let's see. This is again from the chapter Cell, the Unit of Life. This is from the concept plus, what is that? The plastids, right? Chloroplast, vagera. Then if you see, the plastid that stores the xanthophyll is known as, okay, amyloplast. Amyloplast will store, what is it? Starch, isn't it? Chloroplast, right? Chlorophyll A pigment, okay? And aleuroplast, which basically stores, what is it? Hannah. And when you talk about chroma, chromo, colored, right? So the xanthophyll answer is the chromoplast. Okay, moving on to the next one. Smooth endoplasmic reticulum. Again, this is from the same chapter, Cell, the Unit of Life. Okay, right? About the endoplasmic reticulum, we know we have got the rough endoplasmic reticulum as well as the smooth endoplasmic reticulum. So if we talk about this, he asked about the smooth endoplasmic reticulum. It is smooth because it doesn't have the ribosomes. Hai na? If the ribosomes are present on its surface, we call it as rough endoplasmic reticulum, which is responsible for the protein synthesis. So smooth endoplasmic reticulum is a site for the synthesis of carbohydrate. No. Has ribosomes attached to the surface? No. If the ribosomes are attached, we call it as RER. It's the major site for the synthesis of the lipids and it is actively involved in the protein. Actively involved is RER and smooth is involved in the synthesis of the lipids, right? So this is the correct answer. So smooth endoplasmic reticulum is a major site for the synthesis of the lipids, right? So third. So here I have completed the first 15 questions, right? As I've told you, all these questions are straight away from the NCERT, that is 11th standard one, right? Now I would like to call upon or invite Aisha ma'am to carry the next questions, right, of the botany. Thank you, dear students. This one I'll take.
Good evening, everyone. I hope. Good evening, everyone. I hope all you did well in your NEET exam. So we will start from the second half of the botany. So this question is given. The question is given that which of the following character of prokaryotic cell? Basically, this question came from your chapter biological classification, as we discussed that most of the question that came from the 11th part. So this question came from the biological classification. Now, prokaryotic cells means we know they have 70S ribosome. They have 70S ribosome. Okay. So 70S ribosome means we can tell that 70S ribosome, one larger subunit, one smaller subunit will be there. The larger subunit is 50S and the smaller subunit is 30S. So first statement is correct that they have larger subunit and smaller subunit, that is 50S and 30S, which is correct. They can have plasmid. Plasmid, we know that extra chromosomal DNA is there, that is plasmid. So this also they have. Then they contain the mesosomes. Cell membrane enfolding we are seeing in the prokaryotic cell, like inside the cell membrane enfolding we are seeing, which is containing the respiratory material and responsible for the respiration. So that is also there in case of your prokaryote, but they don't have peroxisome. Peroxisome is a cell membrane, your cell organelle, membrane bound cell organelle, which is prokaryote don't have. So we will see which the prokaryote have. Now A, B and C only. So answer will be first option. So this chapter is coming from which chapter? Now biological classification. Okay. Moving forward to the next question, we have match the following. Now Krista, if you will see Krista, what we are telling that if suppose mitochondrial outer membrane is there, the inner membrane is enfolding towards the matrix, and that folding is called as Krista. So enfolding of here mitochondria. This one actually enfolding of mitochondria. Okay, so first one is second option. Next we will go cisternae. Cisternae is, we know that disc shaped sacs in the Golgi apparatus. So answer will be B option will be IV. Then thylakoid, we know that in chloroplast the disc shaped structure will be there, that is called your thylakoid. So thylakoid is present in the flat membrane structure in the stroma of chloroplast. Okay, so this one is A. Now phospholipid, we know that cell membrane is double membrane bound structure, which lipid bilayer structure you are telling. So lipid bilayer structure means this lipid bilayer, which lipid is present? Now phospholipid is present. So phospholipid is present where? Now in the cell membrane. Okay, so here some mistake is there. So this is cell membrane. Okay, so answer will be phospholipid is cell membrane. Now see, first option is second, so A will be second I, then B is IV, B is IV, C is 1 and D is third. So answer will be third option. And which chapter this question came? Now cell the unit of life. Okay, next we will go to the third question, that is what? Now which of the following statement regarding the photorespiration are correct? Okay, so here it is correct. Now, if I will tell that for this chapter is coming from which chapter? Now, photosynthesis in higher plant, which is 12th chapter in your syllabus. Now, see, if I will tell that photorespiration do not occur in the C3 plant. C3 plant characteristics, if you will tell me, one of the characteristics is photorespiration. So, photorespiration doesn't occur in the C4 plant, but it occurs in the C3. So, this one is wrong. Next is oxygen is consumed and carbon dioxide is consumed and oxygen is generated, but in during the photorespiration, as we already know that photorespiration is a waste process for the plant. So during that time what will happen, instead of taking the carbon dioxide, the plant will take the oxygen and producing the carbon dioxide. So this is also wrong, that here the answer will be what? The carbon dioxide is generated and oxygen is consumed. So this is wrong. Next, phosphoglycolate is formed. Phosphoglycolate, when we are telling that Rubisco, RUBP is reacting with oxygen, that time only your photorespiration is occurring. So what is the resultant product? Now 3-phosphoglyceric acid and 2-phosphoglycolate. 2-phosphoglycolate is producing, so this is correct. Next is there is no synthesis of ATP and NADPH. So this is also correct, because during photorespiration there is no synthesis of any form of energy. That's why we are telling that it is a waste process for the plant. So this is also correct. Now, which two are correct in answer? C and D. So, answer will be third option, C and D. And this chapter is your photosynthesis in higher plant. Okay, then we will move further the next question. Next question is coming from the living world, very easy. Now see, arrange the following taxonomic category in the ascending order. Now see, So this is which chapter? Now living world, the first chapter. Okay. Now we know that one mnemonics is there, keep pot clean or family get sick, right? So we are telling, it is telling what? The ascending order. Ascending means lower taxonomic category to higher taxonomic category. So first one will be species, second will be genus, third will be family, then order, then class, then phylum, and lastly kingdom. Right? Now arrange here. First is species. So species is G. Next is genus C. Genus is where? Now here genus A. Then genus after that family. Family is C. Now if we will see after family order. So order is GAC. Okay. So here again order is also in the C they gave. Then after order, class is in the B. Then after that, phylum is in D, and kingdom is in F. So answer will be second option. Okay. Next question. Select the correct sequence of experiment that lead to the gradual understanding of photosynthesis in green plant. So this question also came from photosynthesis in higher plant. Okay. Now, if we are talking about photosynthesis in higher plant, so first your light will absorb by LHC, light harvesting complex. After that, light reaction will occur. Formation of ATP and NADPH will occur. After, by utilization of the product of light reaction, dark reaction is occurring. So see here, first absorption of spectra means by chlorophyll A, chlorophyll B, xanthophyll, all are cheap photosynthetic pigment as well as accessory photosynthetic pigment. The light will absorb first. After that, what you are saying, that production of glucose, this is occurring during the dark reaction. After that, the release of oxygen. Okay. Now here, answer will be which one? The second option. Okay. Next. Endomembrane system include. This question came from your cell the unit of life. Okay, cell the unit of life, this question. Now endomembrane system, we know we have total 4 number of cell organelle which has single membrane bound, and they are coming under the endomembrane system. Why we are telling them endomembrane system? Because they are working coordinatedly, means their one function is related to another function. The other name we can tell, that is GERL system. Right? Now in GERL system, which cell organelle is there? First is endoplasmic reticulum will be there, then Golgi apparatus will be there, then lysosome and vacuole. So now see the answer, endoplasmic reticulum, Golgi apparatus, lysosome and vacuole. So answer will be second option. Next question, length of the stem at time 0 is 20 centimeter and the arithmetic growth rate is 30 centimeter per day, and what is the length of the stem at the end of the 7th day? So this question came from your plant growth and development. We know there are two type of growths are there. One is arithmetic growth, another one is geometric growth. For arithmetic growth, a constant growth will occur at the if you will see the graph of arithmetic growth, it will be a linear graph, right? So what is the formula of arithmetic growth? Now LT is equal to L naught plus RT. Right? So here they gave that time 0, the length is 20 centimeter. That means what? The initial length is 20 centimeter. So L naught is equal to 20 centimeter they gave. Right? Next is the growth rate. Arithmetic growth rate is 30 centimeter. That means R is equal to 30 centimeter per day. Okay, per unit time means per day the growth is 30 centimeter. Now what is the length of the stem at the end of 7th day? So T also they gave us 7th day. Okay, now we have to calculate what? Now LT we have to calculate. So if I have to put all the things which are necessary, L naught is equal to, see, L naught is equal to 20. Now this R is not for 1 day, this R is for 7 days. So now if I will calculate RT, that means what? Now 30 into 7, so 210, right? Now if LT is equal to what now I say? Now L naught plus RT. So L naught is what? Now initial growth that is already given 20 centimeter plus RT, how much I got? 210. So total growth will be 230, right? Answer C, 230 centimeter. So answer will be fourth option. This question, which chapter? The plant growth and development. Okay, next question. This is also again another matchmaking questions, and this questions came from your biological classification kingdom Monera, and we are studying that according to the shape of the particular bacteria we have different type of bacteria, coccus, bacillus, vibrio, spirillum. So that is only came. So this chapter name is biological classification. Okay, now see spherical. Spherical is which one? The coccus, we know coccus is spherical type of bacteria, like this you are telling the streptococcus pneumoniae, right? So coccus means spherical. Next is rod shape. Rod shape is bacillus, example you can tell lactobacillus is a rod shape bacteria. Then comma shape is vibrio. Vibrio cholerae, the comma shape type of structure in the bacteria you are saying. Next is spirulum. Spirulum is spirulina. Now see, A is second, A is second. Next, B is IV. B is IV where? The first option. So answer will be first option. Okay. Next, again another one. The again your matchmaking questions, and this question came from your living world. Okay. This time I told you that living world 3 questions came. So this is the second questions. Now living world, we just discussed one, that species, genus, family, order, kingdom. So now we have to see that which one is family. In living world, you read that family, the suffix will be what? Now major of the family, ACE, AE will be the suffix. Now see here that Anarcardiaceae, the last suffix is ACE, AE, which is the family of mango. Okay, so family is third one. Next, genus. We know that the scientific name of mango is what? Mangifera indica. So Mangifera is the genus. Now genus will be Mangifera. Class, if you will see, monocot or dicot, you are telling that is the class. So class will be dicotyledonine. Phylum, phylum is, it is angiosperm, gymnosperm, bryophyte, pteridophyte. These are my phylum. So phylum will be angiospermy. And order is what? Now we know that order, most of the order, the suffix will be ALES. So answer will be, your order is Sapindales. Okay. So if you will see that A is third eye, A is third. Only one option is there where the A is third. So answer will be first option. Next. Okay. This is a statement based question. This question came from cell cycle cell division. Okay. Cell cycle cell division. Okay. The statement 1 is chromosomes are fully condensed at the end of prophase 1, which is correct. The chromosome is condensing and you can able to properly visualize of the chromosome. So this is correct. Next is meiosis 1 is resemble to mitosis, which is wrong. We know that meiosis is reductional cell division. So the number of chromosome will reduce. Suppose if I am taking 2n number of chromosome, after the meiosis 1, what will be? It will be n number of chromosome. So which is not true. If instead meiosis 1, they will give meiosis 2, then it will be correct. Why? Because meiosis 2 is similar to mitosis, which is a equational cell division. So here statement 1 was correct and statement 2 is wrong. Statement 1 is correct and statement 2 is wrong. So answer will be fourth option. Next, again another matchmaking question which is coming from your morphology of flowering plant. So this chapter is morphology of flowering plant. Now we know placentation, how the ovules are arranged in the ovary, that is called placentation. So according to that we have 5 different type of placentation. Now first one they are giving that where you are seeing the marginal placentation. This is the marginal placentation, right? Which particular plant you are seeing the marginal placentation, the pea plant you are seeing the marginal placentation. Next, axial placentation. Axial placentation, you are seeing what the septums will be there. Then your ovules are arranged in the center. Like, so that you are seeing in the Solanaceae family, so tomato. Next, parietal placentation you will see where na argemon and your mustard you will see. Then free central placentation you will see in the primrose. Okay. Now see, marginal placentation is 4, so I is 4. Next, axial placentation is 2, so B is 2. So answer will be third option. Symbiotic association of fungi and algae. They are fungi and algae, they will do the mutualism. We know that fungi part of the lichen, we are telling what? Now mycobiont, algal part of the your lichen is called as phycobiont. So if I will tell fungi and algae, they are doing the mutualism to form what? The lichen. So answer will be second option. Which of the following is not a prokaryote? Okay, so bacteria is coming under prokaryotic cell kingdom Monera. Then blue-green algae, your cyanobacteria, BGA. So that is also coming under your kingdom Monera. So this is a prokaryote. Mycoplasma, cell wall will be absent, but the smallest organism which is coming under prokaryotic cell. So this is also kingdom Monera. But fungi is multicellular. Fungi is kingdom fungi. You are seeing that is a multicellular. So multicellular will not come under the prokaryotic cell. So this is a eukaryotic cell. So answer will be first option. Next, which of the following characteristics feature is Solanaceae family? This is also another question from the morphology of flowering plant. Now these questions they are giving which one is the Solanaceae family, all the characteristics, and which one is not. Now if I will see flowers are bisexual and actinomorphic, which is correct, which is the characteristics of Solanaceae family. Flowers are bisexual as well as actinomorphic, means in any direction you can cut the flower. So this is correct. Next, calyx are 5 and sepals are united. This is also correct. In case of Solanaceae family, that is a pentamerous flower. So this is also correct. Next, androecium have 5 stamen and epipetalous. Epipetalous condition means what? The androecium is joining to the petal. So this is also correct. Now if you see, ovary is inferior. We know Solanaceae family ovary will be superior ovary, not inferior ovary. Okay. So answer will be A, B and C will be correct. Remaining last one will be wrong because ovary is, in case of Solanaceae family, it is superior, not inferior. Okay. Next, the correct sequence of adult cell cycle phases. In any cell cycle phases, whether we are talking about a fetus or adult, it will be, the sequence will be G1, then S, followed by G2 and M. So answer will be G1, S, G2 and M. 4. This question is coming from your cell cycle cell division. Okay. Assertion is, in an experiment, Mendel observed that F1 progeny plants are all tall and none of them are dwarf. This question first of all came from your principle of inheritance and variations, and this is the 12th chapter. So assertion as they gave that Mendel observed in monohybrid cross, Mendel you are seeing when we are doing suppose the cross, we are taking the true breeding plant. So capital T and small t. If it is forming the gamete, here capital T, here small t. Then we are getting the F1 progeny. F1 progeny we are getting all tall and these are heterozygous tall. So on the basis of this only Mendel gave the law of dominance, that is your one allele will be dominant over another allele. So this is in F1 progeny we are getting all tall. Now, the reason they gave that stem height is a contrasting trait with all tall being dominant and dwarf being recessive. So, if we are talking about a pea plant, 7 contrasting character is there. Out of that, stem height will be one of the contrasting character where tall will be the dominant one and dwarf will be the recessive one. This is the reason why we are seeing in F1 progeny all are tall. So, your reason is also correct and assertion is also correct, and reason is correctly explaining the assertion because in reason they gave that tall is dominant over dwarf. This is because of that only in F1 progeny you are seeing all the tall progeny, right? So both are correct and reason is correctly explanation of the assertion. Okay, chapter name is Principle of Inheritance and Variation. Okay. So next of the Other questions will be discussed by Shiny ma'am. So I will like to invite Shiny ma'am to discuss remaining questions. Thank you.
Thank you, Aisha ma'am. Hello students, I hope your exam went well. Now let's discuss the next set of botany questions. Okay, so the first question is arrange the following in descending order of the number of species in the Amazonian rainforest. This question came from the chapter Biodiversity and its Conservation. The question says that the several options or the several organisms are given below and you have to arrange them on the basis of their descending order of number of species. The maximum number of species out of this is for invertebrates, which is around 1,25,000. Okay, so let's try to find out on the basis of this information that we know. So according to this information, only third option has D as the maximum amount, followed by A, C, B and E. Okay, so first one is invertebrates, then we have plants around 40,000 and then so on. So based on the information that we know, the correct answer will be option number 3. Okay, moving on to the next question. Next question is from the chapter Principle of Inheritance and Variation from topic Multiple Allelism. The question is based on blood grouping. The question is asking for a person with blood group O, which of the following is not possible combination? So if when we will see the options, option number 3, 4 and option number 2 as well shows I gene present as common in the parents. So when you know already for the child to be O blood group, I gene should be there. Both the alleles should be recessive. So option number 3, option number 2 as well as option number 4 is wrong and option number 1 will be the correct answer where the father has IAIB gene and the mother has IAI gene. So when it is crossed, you will see it will be IAI A gene, IA and I. Next, IA and IB and IB and I. So the individual or the child will be blood group A. Blood group A, it can be AB as well as B, but not O blood group. So the correct answer will be option number 1. Moving on to the next question. The next question says, which of the following plant produces non-albuminous seed? The question comes from the chapter Sexual Reproduction in Flowering Plants and this is a direct question which is saying that which of the following is non-albuminous seeds the plant produces. So we know the answer will be P, option number 1. Now the next question is the given below are two sets of statements. One is labeled as assertion and the second is labeled as reason. The first is assertion. Abingdon tortoise in Galapagos Islands became extinct within a decade after goats were introduced. Reason, goats were more efficient at browsing than Abingdon tortoise. Both statements or assertion is true and reason is also true. So the correct answer will be assertion and reason are correct and R is the correct explanation of A. The chapter from which this question comes is biodiversity and its conservation. Next question, which of the following is used as a blood clot buster? So the answer for this question, you know it is a direct question from microbes in human welfare, topic bioactive molecules and the answer will be streptokinase. Next question, which of the following in female gametophyte of an angiosperms helps in guiding the pollen tube for fertilizing the eggs? The questions are directly coming from NCERT, dear students. This question is from the chapter Sexual Reproduction in Flowering Plants and the answer to this is option number 3, synergids. Next question, which of the following statements about lac operon is correct? So this question comes from the chapter molecular basis of inheritance and it is asking about the lac operon. So first is galactose can act as an inducer of an lac operon. It's wrong. Allolactose or lactose acts as a inducer. Second is gene I is constitutively expressed, means it is continuously expressed. Yes, it is correct. Next, lactose activates repressor to bind to the operator. That's wrong. Lactose will inactivate the repressor. Next, fourth option is gene I, Z, Y and A share single common promoter. This is also wrong. The promoter for gene I is separate and for gene ZYA is separate. So the correct option will be option number 2. Next question, which of the following are primary consumers or in a food chain? So the primary consumers in the food chain is herbivores and this simple question comes from the chapter ecosystem. Now let's move ahead to the next question. Next question is, given below are two statements, one is labeled as assertion and the other is labeled as a reason. So the assertion is, the logistic growth model of a population is considered more realistic than the exponential growth model, which is correct. Reason says that resources are finite. So the assertion is explaining us why the logistic growth is more real. Why? Because in general the resources are determined or it is finite in number. Hence, the correct answer will be assertion and reason are correct and R is correct explanation of A, which is option number 2. The question comes from the chapter organism and population. Now moving on to the next question. Next question is a matrix match question from the chapter organism and population. The question says the list 1 shows both species benefited. Next, one species is benefited, other will be okay. First, let's see option number C. Both species are benefited. Both species are benefited in mutualism. One species is benefited while other has no effect on it. Commensalism. Both species are harmed in competition and one species is benefited and other is harmed in predation. So the correct answer will be option number D4, A, C and B. Option number A3, B4, A3, B1. Option number 1 is correct. Now let's move on to the next question. Next question is from the chapter Sexual Reproduction in Flowering Plants. The question is as follows. In the diploid chromosome number of a typical angiosperm is 36. What would be the chromosome number in its endosperm? They are saying in angiosperm, which is diploid, the chromosomes is 36. So N will be here 18. Now asking about the chromosome number in endosperm. So you know in angiosperms the endosperm is triploid in nature. So it will be 3 into 18 which will be 54. Correct answer will be option number 4. Next question. Next question is from the chapter Molecular Basis of Inheritance. The question is, which of the following enzyme synthesizes precursor mRNA? This is also a very direct question from this chapter and the correct answer is RNA polymerase 2. Okay. Next question. This question is also from the chapter Sexual Reproduction in Flowering Plants and again it is a direct NCERT based question which asks how many theca are present in each lobe of a typical bilobed angiosperm anther, and the correct answer is 2. Each lobe contains 2 theca and that's why it is called as bilobed. Next question, which of the following statements are correct? The question comes from the chapter Ecosystem.
which of the following statements is correct? First is energy flow from producers to consumers is unidirectional. This is correct. Next statement, energy pyramid can never be inverted. This is also correct. Next statement, transfer of energy follows the 1% rule. This is wrong. It follows 10% rule. So, statement A and statement B is correct, which makes option number 3 as a correct answer. Next question, or the last question of botany. The question comes from the chapter Principle of Inheritance topic, genetic disorder, and the question gives us two statements. The statement 1 is Down syndrome is caused by the absence of one X chromosome. This is wrong. Down syndrome is caused by trisomy of 21st pair of chromosome. Next statement, Turner syndrome is caused by the presence of an additional copy of the chromosome. Statement 2 is also wrong, beta. It is caused by the absence of one X chromosome, and Turner syndrome is seen in females. So both the statements are incorrect, and the correct answer will be option number 3. Now that's all for botany. Now I'll hand over to Mahima ma'am for zoology discussion.
Hello students, I hope you have written very well. Okay, so the paper overall in the zoology was directly from NCRT based. Okay, you can see the direct questions from the NCRT lines, and they were mostly the direct questions. So when comparative to the last examination, here in this examination, the number of direct questions were more, and they were completely based on the NCRT. Now let us quickly start the paper discussion for zoology. Yeah. Yes, so let us read the first question. Now here the first question is the statement based. Now let us read the first statement. Okay, so statement 1, the class Reptilia refers to creeping or crawling mode of locomotion. Obviously. So why do we name them reptiles? Because they creep and they crawl. Next, all organisms belong to Reptilia have three-chambered heart. Okay, so do all of them they have the three-chambered heart? It's no. Okay, most of them they have a three-chambered heart, but we have an exception like crocodile. Okay, so what we can go, the first statement is correct, but the second statement is what? Incorrect. So what will be the right answer? Okay, the first statement is correct, but the second statement is incorrect. So we can go with the option number 4. Next. So let us go to the second question. So here it's a direct question again. So what they are asking, which of the following represent the correct sequence of arrangement of the bone in the lower limb of the human? So what exactly the bones we have? So we have the longest bone, femur, followed by patella. Okay, so let us see the sequence. Okay, then here what do we have? First we should look for femur. So here the remaining femur is just given, here patella. The femur is followed by what? It is followed by the patella. So here you can see, you can just cross the remaining options. Fine. Now here femur, patella, then we have tibia, then we have the tarsal bone. Okay, so this is, here we can see the correct sequencing of the order of the bones there. So we need to go with the option number 4. Let us go to the next question. In water frogs respire using? So here again a direct question from the NCRT. So we know that the frogs, when they are in the water, they respire via what? Their skin, and that type of respiration is known as cutaneous respiration. Okay, when they are on land, they can go with the, buccopharyngeal, they can go with the pulmonary, right? But when they are in the water, they are exclusively what? The skin for the respiration. So again a direct question, so we can go with the option number 2. I hope you have marked it correctly, students. Now let us go to the next question. So in the next question, which of the following is not a characteristic of the chordate? So we all know the characteristic of the chordates, right? So here we need to select for the one which is not a characteristic. So what is the first one? Presence of post-anal tail. That's right, they do have a post-anal tail. Next, presence of notochord. Yes. Now central nervous system is dorsal. Yes, it is. Next, the absence of gills. Are gills absent? No. The gills are present there. So here we need to go with the one because they are asking for not a characteristic. So here the correct option will be what? The correct option will be the option number 4. Now let us go to the next question. Now which of the following statement is related to the pituitary gland and is correct?
Now here it is divided into anatomically into adenohypophysis and neurohypophysis. As we all know, the pituitary gland, it is actually divided into what? Okay, the adenohypophysis, and then we have a neurohypophysis. Next, it secretes the follicle stimulating hormone. That's right, it releases the follicle stimulating hormone. Next, it secretes melanocytic stimulating hormone. Yeah, it also releases a melanocytic releasing hormone. Next, it does not secrete prolactin. Is it? No. It also secretes what? The prolactin. So as they are asking for the correct option, so we can go with the option number A, B and C. So the correct answer will be the option number C there. Okay. Now let us quickly go to the next question. Now which of the following statement is incorrect? We need to look up for the statement which is incorrect. So here the first question, fibrinogen is produced from the fibrin, is it? So when we see this entire mechanism, so here fibrinogen is converted into the fibrin, but here in the statement they are saying what? Fibrinogen is produced from fibrin, which is absolutely wrong. Next, blood coagulation is response to an injury. Yes, that's right. Now next, blood clot consists of the fibrins. Yes. Now next, fibrin is produced from the fibrinogen. Absolutely. So fibrin is actually converted into the soluble fibrin. Okay. So here this is the one. So we need to go with the incorrect. So option number 1 would be the right answer. Now next one. Yeah, now here we have a matrix matching. Again, you know, if you remember the biomolecules chapter, we have a table. Okay, so this is from the secondary metabolite one. This question is from the chapter biomolecules. So now here, the starch. What exactly is a starch? It is a energy storage. Okay, when coming to the next one, that is antibody. Now what is the function of the antibody? The antibody helps in what? The fight against the pathogen. So here, the fight infection. Next, what do we have? The concavalin A. We know it is what? The lectin. Next, we have GLUT4, which is again a glucose transport. So, what will be the answer? The A is 2, okay, B is 1, and C is your 4, and D is your 3. So, option number 3 would be the correct one. Next, moving forward to the next question, again a direct question from the NCERT. Now here they are asking the number of vertebrae in the human. It's a direct question, students. We all know that it consists of what? 26 vertebrae. Okay. So the answer will be what? Option number 4, and this question is from the chapter locomotion and movement. Now moving forward to the next question, the number of the action potentials. Okay, so as we know, the number of action potential generated by the sinoatrial node in a healthy human is. We know that for generating the impulses, the sinoatrial node will be acting like a pacemaker. So the number of, they will be actually generating the impulses. Okay, so here the number of action potential. So one action potential is equal to one heartbeat. So how many do they generate as they are the pacemaker? Again a direct line from the NCERT. It is none than 70 to 75. So we can go with the option number 3. Okay, next. Now, which of the following is not a part of the human central neural system? Now, again a direct question, students. Now, this question is taken from the chapter, the neural control and coordination in the brain part. So, we know we have this meninges. Okay, so the meninges are the layers which actually cover. So, what do we have? We have 3 meninges. So, let us see here. We have the pia mater. Okay, here they have given pericardium. We all know the pericardium is not, that is actually the covering there. Okay, it is the covering of the heart. Okay, when we talk about the next arachnoid matter, dura matter, pia matter, so they are the meninges which are present in the brain. Okay, so here is the question they are asking which is not a part. So we can go with the option number 1. Okay, next. Now arrange the following elements in descending order of their contribution to percentage weight of the human body. So this question is taken from the chapter biomolecules, again a table. Okay, so here we know that the oxygen, okay, is 65%, fine. The carbon consists of 18.5%, right? Then we have the hydrogen, okay, they are talking about the hydrogen which is 9.5%. Next we have this nitrogen which is 3.2%. So they are asking descending order. So what need to be the sequence, students? The sequencing should be first oxygen followed by the carbon, okay, then hydrogen, then nitrogen. So it will be A, B, C and D, then we can go with the option number 2nd. So direct question from the NCERT, students. Fine. Next, let us move to the next question. Which of the following statement is incorrect? So this question is taken from the chapter chemical control and coordination from the part pancreas. We know that the pancreas is a heterocrine gland. okay, which is having the exocrine part and the endocrine part. In the endocrine part, we have the islets of Langerhans, which have two types of cells, alpha cells and beta cells. We know alpha cells releases glucagon and beta cells releases what? Insulin. Okay, so let us read out the statement. So first they are saying beta cells of pancreas secrete insulin. Yes, alpha cells secrete glucagon and beta cells releases what? The insulin. Second, alpha cells produces glucagon. That's right. Third, alpha cells secrete the insulin? No, it doesn't. Okay, it doesn't secrete the insulin. Next, glucagon stimulates glycogenolysis. Yes. Now, what is the glucagon? When we talk about the glucagon, it is a hypercalcemic hormone. It is hyperglycemic, it is hyperglycemic hormone, so it increases the blood sugar level. How does it increase the blood sugar level? It acts on the hepatocytes and increases the blood sugar level. So here the answer will be what? As they are asking for incorrect, the answer will be option number 3. Next, we have the two statements. Statement 1, when any plane passes through the central axis of the body, divides the organism into two identical halves. It is any plane and it is called radial symmetry. That's right. Next, in phylum Echinodermata, both adults and larvae are radially symmetrical. No, we know that the adult is what? The adult is, okay, radial symmetry, and when we talk about the larva, it is bilateral symmetry. It is what symmetry, students? Bilateral. That's the reason we say that the larva is more advanced than the adult. Okay, so first statement is right, but the second statement is what? Wrong. So here what we can say? Yeah, we can go with the option. The statement 1 is correct, but the statement 2 is what? Incorrect. Okay, so here this question is from the animal kingdom chapter. Okay, next let us move on to the next question. In frogs, the number of pair of cranial nerves arising from the brain. So how many pairs we have? We have 10 pair of cranial nerves. Okay, direct line. Okay, from the NCRT. So here we have 10 pair of cranial nerves arising from the brain. This question is again from the topic frog from structural organization in animals. Okay, so the overall questions were directly from NCRT. Right now, for discussing the next set of questions, I would like to call Kavya ma'am to join us.
Hello students, how was the paper? The paper was very easy, right? So we will be discussing about the few set of questions from zoology. Few set were already discussed by Mahima ma'am. I'll be discussing about the next few set of questions. So if you analyze the paper, it was much easy paper directly from the NCRT. The entire bio paper was very easy, right? It was more from human reproduction, reproductive health, and even it was from evolution. It was having a more weightage of the second year topics when compared to the first year topics. Okay, so straight away let's get into the set of questions. The first question, if you see it over here, this is taken from the chapter Biotechnology Principles and Process. So it is an assertion and reason type of question. If you see this, assertion says in recombinant DNA technology, lysozyme is used for disrupting bacterial cell walls while cellulase is for the plant cells. We know it very well that the bacterial cell wall is made up of peptidoglycan, and peptidoglycan, when any bacteria is being taken into our mouth, then the lysozyme, the salivary amylase, is having certain secretions of lysozyme which is antibacterial in nature, which acts on the cell wall, that is the peptidoglycan layer of the bacterial cell, and which breaks it down. So lysozyme is obviously used for the disrupting of the bacterial cell wall, which is actually composed of peptidoglycan, which is actually composed of peptidoglycan. Next, cellulase. The same is the case with the plant cell walls as well. The plant cell wall is made up of cellulose, and the enzyme used to break down that cellulose is nothing but cellulase. So the assertion which says that the rDNA technology, lysozyme is used for disrupting the bacterial cell while the cellulase is used for the plant cell is correct. This is a correct statement. Coming to the reason, Isolation of genetic material need disruption of the cell. The reason is also correct because when we are willing to isolate any genetic material in recombinant DNA technology, the cell wall is supposed to be broken because where is the genetic material present? It is present deep inside the cell in the nucleus. So if we break down the cell wall, then only we will be able to extract the genetic material. So the reason is correct, and you can see that reason is perfectly explaining the assertion as well. So our correct option would be second option. Both A and R are correct, and R is the correct explanation of A. Moving to the next question, this question is taken from reproductive health, from the subtopic that is assisted reproductive technologies, ARTs. So let's see what is the question. The method of directly injecting a sperm into the ovum in an assisted reproductive technology is called. This is the easiest question that could be asked from ARTs. So when a sperm is directly injected into the cytoplasm of the ovum, the technique is nothing but intracytoplasmic sperm injection. It is intracytoplasmic sperm injection, which is done in vitro fertilization technique. For in vitro fertilization, the zygote is cultured in a laboratory condition. In that laboratory, what is done? The sperm collected from the donor is being injected into the cytoplasm of the ovum. So the correct option for this is fourth option, that is intracytoplasmic sperm injection. Coming to the next question, adaptive radiation in placental mammals and Australian marsupials leading to similarity between distant species is an example of. This question is taken from the chapter Evolution, from the subtopic Australia, that is nothing but the adaptive radiation. Adaptive radiation has few examples. The Australian marsupials and placental mammals, and Darwin also told about the adaptations in the Darwin finches of the Galapagos Islands. And when we talk about adaptive radiation, it is nothing but an example of convergent evolution. So Australian marsupials and the placental mammals are an example of convergent evolution. Moving to the next question. This question is taken from the chapter Animal Kingdom from invertebrates part. This is from the Porifera. Sponges exchange O2 and CO2 by. We know that the sponges are having a well-developed characteristic feature. Well-developed characteristic feature. What is that characteristic feature? It is the water canal system, which is also called as the aquiferous system. In this system, they are having the water which is entering through the ostia into the spongiocele and exiting through the osculum. What is the function of this water canal system? Nothing but intake of food, exchange of the respiratory gases, removal of the waste, and transfer of the gametes. What is the second function that we mentioned? It is nothing but the exchange of the respiratory gases. This O2 and CO2 are usually exchanged by the sponges by simple diffusion over the entire body surface. They are having pores over the entire body surface, which is responsible for the diffusion of gases. Moving to the next question. This question is again taken from the chapter Evolution. As I said, many questions are from evolution. So if you see, this is a statement type of question. Statement 1 says, modern Homo sapiens arose in Australia and moved across countries. From where did Australia come? The modern Homo sapiens did not arose in Australia, but they arose in Africa, and then they moved across the other countries. So the statement 1 is incorrect. Coming to the statement 2, Homo sapiens arose around 75,000 to 10,000 years ago. Yes, Homo sapiens were born in Ice Age. They developed, they evolved in Ice Age. So they evolved around 75,000 to 10,000 years ago, which is a correct statement. So in the option, statement 1 is incorrect, but statement 2 is correct. Option 1 is the correct option. Moving to the next question, which of the following is used as the effective sedative and painkiller for the treating of the post-surgery patients? This is the question which have been taken from the chapter Human Health and Diseases, from the subtopic that is drugs, alcohol abuse. So if you see, when they are saying effective sedative and painkiller for treating the post-surgery patients, we know it very well that a drug, morphine, which is extracted from Papaver somniferum, that is a poppy plant, is used as an effective sedative. And for the people who have, for the patient who has undergone surgery, it is given for the sedative or as painkiller as well. So the option 4 is the correct option. Moving to the next question. This question is taken from the chapter Human Reproduction. The covering of the ovum at ovulation. We know it very well that the ovulation, at the time of ovulation, it is usually released in the form of secondary oocyte. This secondary oocyte has a layer around itself. That is nothing but zona pellucida. It is a direct NCERT statement that secondary oocyte develops a layer around itself called as zona pellucida. And during ovulation, the ovum is usually released along with the zona pellucida. So here our option is option 4, which is marked to be the correct one. Moving to the next question, again from human reproduction. Which of the following structure is not a part of the male reproductive system? So if you see the first option, infundibulum. Infundibulum is a part of a fallopian tube. The funnel-shaped structure of the fallopian tube, which is collecting the ova while the ovulation is occurring, is called as infundibulum. This infundibulum is having finger-like projections called as fimbriae. So infundibulum is the part of female reproductive system but not the male reproductive system. Whereas radiotestis, which is an accessory duct, epididymis, which is also an accessory duct, and vasa efferentia, which is also an accessory duct, is accessory ducts of the male reproductive system. Moving to the next question. This question is a statement type of question, again taken from the chapter Human Reproduction. So statement 1 says, ovulation is caused by the LH surge leading to the rupture of the Graafian follicle. This is a correct statement. Why is it correct? Because we know in the menstrual cycle, the 14th day of the menstrual cycle is the ovulatory phase, where because of high amount of estrogen, LH and FSH, the Graafian follicle ruptures and the ovum is released from the Graafian follicle. And that phase we are having the highest amount of LH, which is called as LH surge. So the first statement is correct. Moving to the second statement, Graafian follicle remaining after ovulation transform into corpus luteum and secretes large amount of estrogen. Till here, that means Graafian follicle remaining after ovulation transform into corpus luteum. It is correct. Why? Because Graafian follicle turns into a yellow colored body called as corpus luteum, which is secreting progesterone but not estrogen. It is secreting progesterone but not estrogen. So this is an incorrect statement. Then that yellow colored body transforms into a white colored body called as corpus albicans. So statement 1 is correct, but statement 2 is incorrect. So our correct option is option 4. Moving to the next question, the opening between the right atrium and right ventricle is guarded by. This is a question which is taken from body fluids and its circulation. Now, in this question, it is about the structure of the heart. There are certain AV valves, right? These AV valves are bicuspid valve and tricuspid valve. We know it very well that the tricuspid valve is present at the junction of right atrium to right ventricle, whereas bicuspid or mitral valve is present between the left atrium and left ventricle. So our correct option, because they have asked for right atrium and right ventricle, our correct option is tricuspid valve. Moving to the next question, which of the following is not the evidence for evolution? So here it is, divergent evolution of anatomical structures such as forelimbs. We know it very well. Forelimbs are usually similar in character, similar in structure, but their functions are different, which is an example of homologous organs. And homologous organs show which type of evolution? Divergent evolution. So this is the correct statement. Next, if you see, convergent evolution of traits like wings of bats and butterfly. Wings of bats and butterfly are an example of analogous organs which are different in structure, but their function is very similar, and they are nothing because of the convergent evolution. So both are correct. Statement 1 and statement 2 are because of the evolution, or you can say comparative anatomy. Comparative anatomy. From this branch of science, it is the evolutionary evidence. Next, if you see paleontological evidence from the fossil records, this is the evolutionary evidence from the branch of science that is nothing but paleontology. This is also a correct statement. Actually, this option 4 might be a little confusing. What is it? Embryological support for evolution as proposed by Ernest Haeckel. So if we generally read this option, we will feel it is very true that the embryological support was given by Ernest Haeckel. But point to be noted here, there was a controversy on the theory given by Ernest Haeckel. What was that controversy? There was a controversy that all embryological characteristics are not being seen in the adult forms, or are not being inherited in the evolved forms, which was disproved by Carl von Baer, which was disproved by Carl von Baer. Carl von Baer, he gave another embryology. What is that embryology? That is modern embryology. So here, this is not considered to be the evidence of evolution. Okay, next if you see, coming to the next question, this is again taken from the biotechnology and its application. It is about the improvement, or you can say the use of the RDNA technology or biotechnology in the agricultural sciences. In the inactive form of Bt toxin is converted into the active form in the insect gut. So here they are asking actually about the pH or the enzymes which are present. So we know it very well that in the alkaline gut, that is in the alkaline pH of the gut, the Bt prototoxin is converted into an active form. This active form will kill the insect and it will affect the insect and protecting the plant. So this is what is due to the alkaline pH of the gut of the insect. Next, coming to the question, this is again taken from Human Health and Diseases from antibodies. The colostrum secreted by mother during the initial stages of lactogen is abundant in. Usually, we know there are different types of immunity. The different types of immunity include active immunity and passive immunity. Active immunity is again natural and artificial. Passive is again natural and artificial. In natural passive immunity, we learned two examples. IgA type of antibodies are being transferred while breastfeeding from the mother to the baby, which is increasing the resistance power in the baby. So IgA type of antibodies are present in the initial milk, the yellowish fluid milk of the mother. Which antibodies is it? It is IgA type of antibodies. Coming to the last question, which of the following disease is not sexually transmitted? So we have to go it one by one. Genital warts. The genital warts is caused by human papilloma virus, which is a sexually transmitted disease. So this can be eliminated. Syphilis is caused by Treponema pallidum, which is also a sexually transmitted disease. This can also be eliminated. Third option is tuberculosis, which is caused by a bacteria, Mycobacterium tuberculosis, which is not a sexually transmitted disease. If you see gonorrhea, gonorrhea is also caused by Neisseria gonorrhoeae, which is again a sexually transmitted disease. So which of the following is not a sexually transmitted disease? It is nothing but tuberculosis. So I hope the questions that I have discussed, you have marked them correct. The next set of questions will be now taken up by Bhavna.
Very good evening, all of you. This is Dr. Bhavna, Zoology faculty. Today we are going to deal with the next set of questions. Hope today biology paper is very easy and next set of questions will be continued by me. Let us go into the next set of questions. And the first question is from biotechnology. So in biotechnology, this is a match the type of following question. And here the transformation. So the transformation basically starts with which type? Transfer of DNA to the host bacteria. So this is 2. And cloning site. Cloning site is where the restriction enzymes basically cut. So this cloning site is associated with the restriction enzyme. Next we have selection. Selection means it is nothing but the selectable marker. So the selectable marker is associated with the antibiotics. And we have the ORI site. ORI site means by name only you can understand that ORI site is origin of replication. So it is 3. So we can go directly with the match the following type of question as 2, 1, 4, 3. So the best suitable option is second option. Next question. A population of diploid organisms is at Hardy-Weinberg equilibrium if the frequency of allele A is 0.1 they have given. So allele A is 0.1 and this question is from evolution topic. So frequency of double A. So A square they are asking. So directly 0.1 into 0.1. We get 0.01 as the answer. So the key is second option. This is a direct question from evolution. The next question, sperm motility is due to. So this sperm motility is a question from locomotion and movement. We have 4 types of movement. One is muscular movement, flagellar movement, ciliary movement and amoeboid movement. But sperm motility or protozoans like euglena, everyone exhibit directly flagellar movement because the tail moves. So it is a flagellated movement. So direct option we can go with flagellar movement. Next question, given below are the two statements as assertion and reason. Last time NEET they have not given assertion reason type of questions in bio, but this time assertion reason questions are given. And the assertion is forelimbs of human and bats are homologous. So this question is again from evolution chapter. Homologous means they have same structure but different function. So what is the reason they have given? Forelimbs of humans and bats have similar anatomical structure and same origin also they have, but different functions are there. So we can directly go with the assertion and reason are correct. So both assertion and reason are correct and R is the correct explanation of it. But the options they have changed. So we can go with the second option. Next question, muscle contraction is initiated by a signal sent by the CNS by the release of. So this question is from locomotion and movement chapter. Directly see the muscle contraction will start whenever from the motor neuron to the muscle the impulse is transferred. But from neuron to muscle when the impulse is transferred, and this is through a neurotransmitter which is present. What is the neurotransmitter? Direct question, and it is acetylcholine is the neurotransmitter. Is it excitatory or inhibitory type of neurotransmitter? This is an excitatory type of neurotransmitter, so the muscles will get activated. The next question, which of the following hormone is not secreted by human placenta? This is a direct question from human reproduction. So placenta will secrete HCG, HPL, estrogen, progesterone hormones. But what it won't secrete is LH. LH is basically secreted by anterior pituitary gland and it will secrete testosterone and all. So human placenta won't secrete, they are asking. So it is LH. Next question. Which of the following statement is correct about Plasmodium species? Same Plasmodium type of question has been repeated and it is from human health and diseases. So this question is related to malaria because malaria is caused by Plasmodium species and where the fertilization takes place. Correct statement. So fertilization takes place in the mosquito gut. Gut will help in the process of fertilization because fusion of male and female gametes will occur in that place. Then reproduce sexually in liver cells is wrong. They reproduce asexually. And the third option also reproduce sexually in RBC is wrong. Asexual reproduction is seen in liver cells as well as RBCs. Next, gametocyte develop in the mosquito gut is wrong. It will develop in humans. So which is the correct statement is fertilization takes place here. Then, which of the following statements about the reabsorption process in Henle's loop are correct? So, this is from excretion chapter And we have like statement questions. So the descending limb is permeable to water but impermeable to electrolytes. Yes, the descending limb is permeable only to water. Means water will be coming out but impermeable to electrolytes. It's mostly reabsorbed there. Ascending limb is opposite to that. So this statement is correct. Urine gets concentrated in Henle's loop because countercurrent mechanism and all is associated with Henle's loop. So urine gets concentrated here. Next, reabsorption of sodium and water take place in Henle's loop. According to NCRT, reabsorption of sodium and water take place only in DCT. So this statement is wrong. Next, active or passive transport of electrolytes occur in ascending limb of loop of Henle. Yes, active transport may be there or passive transport. Active means with the use of energy. Passive means without the use of energy it will take place. So what are the statements which are correct are A, B and D. The next question is from locomotion and movement. Which of the following is correct order of arrangement of vertebrae? They have given see from head to toe region. So from head to toe means again we have to consider the vertebral column where cervical vertebrae, then thoracic vertebrae and then lumbar and finally it is sacral vertebrae. After sacral also there is coccygeal vertebrae. So this is in an order. They're asking in a sequence. So cervical which are 7, thoracic are 12, lumbar are 5, sacral 5 used to form in 1 in adults. So this is the arrangement which is the first option. Given below are two statements. So this is a statement type of question from biotechnology applications. Plasmids are autonomously replicating DNA. Means plasmids have the capacity to self-replicate the DNA. So this is the correct statement. Next statement of plasmids are extra chromosomal DNA. Yeah, they are extra chromosomal and they are circular DNA which will help as vectors. So both the statements are correct. So we can go with the option as both 1 and 2 are correct. Next question is from evolution. Natural selection can lead to. Natural selection is stabilization. See, we have the graphs. How the natural selection will help. This is stabilization and we have directional change. Means towards one side the graph is shifted and the third one we have is disruption where the two peaks are calculated. The middle part is not calculated here. So here the only middle part, that's why stabilizing selection. Here towards one side it moves and here there are two extreme ends are selected than the middle population. So it is stabilization, directional selection and disruption. Genetic drift is not associated with natural selection. It is also another factor which will affect the Hardy-Weinberg equilibrium. So we can go with the options as A, C and D. Next is the match the following type of question and this is from chemical control and coordination. So this chemical control, what about the hormones function they are asking here. So excess growth hormone. Excess growth hormone leads to a disease in adults which is only called as acromegaly. And then we have about LH. As previously we discussed, LH will play an important role in ovulation, release of ovum. Then we have vasopressin. Vasopressin also called as ADH. So it will help in reabsorption of sodium and water. And then we have oxytocin. Oxytocin help in contractions of uterus during the childbirth and basically help in movements. So this is associated with this. So 3, 4, 1 and 2. So we can go with the third option. 3, 4, 1 and 2. The next question is, which of the following are secondary lymphoid organs? This question is a direct from human health and diseases. So they are asking which are lymphoid organs. Obviously, we know bone marrow and thymus both are primary lymphoid organs and we have tonsils and spleen which are secondary lymphoid organs. What is the use of secondary lymphoid organs? Are they help in differentiation and interaction of lymphocytes with the antigen. So we have the options as only B and C. Next question. This question is from biotechnology. During PCR, primers bind to DNA strands in the which step they are asking. So we have three steps basically. First is denaturation. Then we have annealing, and the final step is extinction. So denaturation, annealing and extinction. In the first step of denaturation, we basically not add the primers, just we will separate the DNA strands. And in the second step, which is annealing process, new primers are added, and in extinction process, we will extend the primers. So the answer is annealing, because they are asking the question as primers bind to DNA in which step. So this is about the annealing step. So we are done with the zoology part. Hope it is very useful. And next, Pujita ma'am will continue with the physics part.
Hello students, I hope you have performed well in your exam. So don't worry, I understand the paper is a little bit difficult, but fear not, we are here to explain you every question in detail. So let's start with the first question. So we have a question from dual nature, and the question goes as, a photon and an electron have the same energy. So what will be the formula of energy and photon relation? We know P is equals to under root 2mE, and at the same time we have the formula for momentum as E by C. Which one is for photon and which one is for electron? This is for electron because it has some mass, whereas for photon, it travels with the speed of light, so this will be the formula. We just have to find out the ratio of these two. Let's first find out what is the value of it. Under root 2, he gave you already mass of electron, 1.6 into 10 to the power of minus 19. Energy, he gave 20 electron volts. Focus on the units. It is 1.6 into 10 to the power of minus 19. So let's do under root of this. Just give me a second. We'll just write the mass of electron here as 9 into 10 to the power minus 31. So let's focus here. 9, 2 into 2, 2 square, 3 square, 4 square. So if you take all this, you will get 24 square. And then 10 power minus 31 minus 19, which gives you 50. So it gives you 10 to the power minus 25. Let's go for the next one. 20 electron volts, 20 into 1.6 into 10 to the power of minus 19 divided by 3 into 10 to the power 8. So this is going to give you 32 divided by 3 into 10 to the power minus 27. So if you do a fraction of these two, you will get the fraction as 24 divided by 32 multiplied by 3 into 10 to the power minus 25 plus 27 plus 2. So we have minus 2 here. Just erase this part and write minus 2. But focus on the units. What are the units here? Units are, for the next one, and you have here the units whole divided by 24 into 3, 72 divided by 32 into 10 to the power plus 2, which is giving you the answer as 225. It's 2.25 multiplied by 10 power 2. Let's go for the next question. And the next question is from fluids chapter. He has given you 2 points P and Q, and he gave you the pressure difference for the 2 points as 50 Newton meter per minus 2, and the cross sectional areas of P and Q individually. He's asking you to find out rate of flow. What is rate of flow? Q is equals to AV. You can find velocity at point P or at point Q. We also know another relation for Q, which is equal to A1 V1 is equals to A2 V2. Since here the area has reduced to half, its velocity must be double than the first case. Now let's use the same relation which I got from the continuity equation, and I'll be putting it back in Bernoulli's theorem. What is Bernoulli's theorem? Half rho 1 V1 square plus P1 is equals to half rho 2 V2 square plus P2. We are not writing rho change because they are going to lie on the same level. Let's go for the next step. These are the points at point P and these at point Q. So pressure difference is equals to half rho into, since the densities are same, VQ square minus VP square. We know VQ is double of it. So 2 square minus 1 will give you 3VP square and delta P. Now let's start solving this. What is delta P value? 15 is equals to 1000, which is the density of the water, into Vp square 3. When it goes here, gives you 30. All the three digits got cancelled. So Vp value, you're getting it as 0.1 meter per second. But you have to answer in centimeter cube second power minus 1. So I'm going to convert this into centimeters. And then I will put it back in this. Since I got velocity of P, I'll write here APVP. AP value is 40, so multiply 40 by 10, and what will be the answer? Exactly 400 is our answer. Let's move to the next question. This is from rotational motion, and here he asked you about the angular momentum. He gave you 3.08d, and he gave you the distance also, but it doesn't matter. Why? When you have a point here and when there is no perpendicular distance, suppose you have a sphere here and its center of mass is also moving, then you will have certain momentum because the total angular momentum depends on the angular momentum which is passed through the center, which is the spin, and the angular momentum which is r bar cross p bar. Since we don't have any momentum, center of mass is 0, this term is 0, so the whole term becomes 0. So at any point on this axis, the momentum has to remain same. So 4 should be the right answer. The next question is from solenoid subtopic. We all know about this, CMI. So let's start with flux. What is the relation N phi is equals to Ni? And we also know the formula of phi. What is the formula of phi? NBA is equals to L divided by I. We know the formula of capital N. Capital N is number of turns. Capital N is number of turns per unit length multiplied by length, which gives you number of turns. Mu naught Ni pi r square divided by the current. When both the currents gets cancelled, mu naught N square L pi r square will be your answer, which is option number 2. Next question. Thermal expansion, thermal properties of matter is the subtopic. Let's read what is the question. Temperature of a metallic sphere is increased by a small amount. When temperature increases and we ask you to find the increase in volume, what is the formula we all know? Delta V is equals to V gamma delta T. What is the volume of it? 4 by 3 pi R cube. Gamma is not mentioned, but he gave you alpha. What is the relation between gamma and alpha? 3 alpha, exactly. Into delta T. Now just cancel the terms here and find out what is the answer. Option number 4. And the next question is from current electricity when he gave you resistors. So let's just use the concept and understand. When you have two resistors connected in series, he's just asking us from the options what happens to the current. And when you have alpha once increasing and the other case alpha decreasing, then the total resistance for the series condition will be added, whereas for parallel, as we know, it should be R1 R2 by R1 plus R2, and since it's just like the reduced mass concept, we already know that the resultant resistance has to be smaller than smaller resistance. This is more than enough once you know this answer. Anytime you get this ratio, the final result will be smaller than the smaller term. Since by passing the temperature, its resistance is going to decrease, overall the resistance will further decrease, so current has to increase. How many options we have? Two options. When it comes to this, while addition, these two terms gets cancelled, so resistance is going to be same. Obviously, current also must remain same. So the correct option is option number 2. Next question is from modern physics, photoelectric effect. Photoelectric effect question. He has told you that the power is decreasing with time. Power is related to intensity. Intensity is related to Intensity is related to. Let me tell you a small trick here. How I used to remember is if you have intensity, remember all things that are I. Similarly, V. Where stopping potential depends on the frequency and which affects the velocity of it. So simultaneously it will affect the kinetic energy also, whereas intensity will be affected by photoelectric current. So these terms are independent of each other, only these are directly proportional. Since the power decreases, obviously it has to decrease its intensity, which decreases the current. Current must get decreased, but it should not affect the stopping potential. Obviously, option number 2. Let's go for the next question. Such an easy one. He has given you the total voltage here. What is the total voltage? Let's just ignore the sign convention as of now. It's 5 volts and the voltage across these two terminals is 3. What will be the remaining part? 5 has to be divided for these two. So the leftover part has to be 2. Next question. This is terminal velocity. So from fluids. What is the formula of terminal velocity? Terminal velocity formula depends on both the densities as rho minus sigma. So if I take sigma common also, I will have rho by sigma minus 1. When it's directly proportional to this, I will be expressing this as y is equal to mx minus c. How does the graph look for it? It should have a negative y-intercept with a positive slope. And where will be it intersecting this axis? That is the point where this whole term has to become 0. When will it become 0? When rho by sigma is equal to 1. And this is the position where it has to become 1. So option 2 is your right answer. Next question, gravitation. Such an easy one. He gave you equal mass and then he gave you the radii ratio here. Escape formula is under root 2 gm by r. So when I use 2 gm by r formula, we know that it has to be inversely proportional to radius with r. Since the ratio of the radii is, let's just go for the next question and show you here. Yeah. What is the ratio of the radii? 1 is to 2. R1 by R2 will be 2 by 1. Why to R1 by R2? Because he asked me V2 by V1. Since I want V2 by V1, I'll just put a root over this, which gives me answer option 4. Next question. Alternating current. Most of the students love this chapter. He has given you omega because that's the coefficient of T. And then he gave you the total voltage V naught value. He's asking us to find the value of current. V is equal to IZ. And what is Z formula? Under root R square plus XL minus XC whole square. So you have to find out what is XL and XC because you already know what is R. XL formula omega L, XC formula 1 by omega C. What is omega value? 2 into 10 to the power 3. L value 10 into 10 to the power minus 3. So obviously it's going to give you answer as 20. Let's go for the next case. Omega is again 2 into 10 to the power 3, but C is 25 into 10 power minus 6. Again it goes there, you're going to get the same answer as 20. When XL and XC both are equal, this term goes to 0, you are left with R. So when Z is R, I will be V by R. Voltage value is 220 and R value is 100. So your I value has to be 2.2. So let's go for next question. Two identical inductors. So this is also from EMI, mutual induction concept. Be careful here, he gave you point A and B here and A and B here, not here. So for these two, you will take the effective self-inductance, which is for parallel, it becomes half when they are identical. Whereas here the inductance is anyway L. So now we are going to find out EMF. What will be the formula of EMF, how does it depend on L? Induced EMF depends on L, whereas phi is equals to EMF is equals to minus d phi by dt. So L into di by dt is directly proportional to L. So whatever ratio you have, L by 2, so it should give you the answer option 1. Let's go for the next question. It's here. Three identical capacitors. So electric potential and capacitance is the chapter where he asked us to find out what is UP and UT. UP is only for the specific capacitor, whereas UT is for the total. For total, you need to use the total effective capacitance into V square, which you already know. How will you find the effective capacitance? These two are in parallel series. So it goes to C by 2, this will be C. Now finally you have to add them, which gives you 3C by 2. And now you have to find out the potential energy here, which gives you with this formula, which is Q square by 2C. What is Q? Q is equal to C. Let's see what will be the charge here. Here the capacitance is C, but the charge is going to be divided as half. So you will have only V is going to be same. Capacitance here is C by 2 and V whole square divided by 2C, which gives you here 1 by 8 ratio and here 3 by 4. When you do a ratio of these two, 4 cancels this by 2 and 3 2s are 6. Answer has to be 1 by 6. Just do a ratio of these two. Let's go for the next question. Yeah, next question is area is changing. So it's asking us to find out what happens to the area value he gave you here. A is equals to A naught into 1 plus sine t. And he said power. What will be the power dissipated with respect to time? How will power change with time? To find out power, we know it is V square by power P is equals to VI. And we know V is equals to IR, or you can just represent I as V by R, so it gives you V square by R or EMF square. So to find power, you need EMF. How do we find EMF? EMF is equals to minus d phi by dt. What is phi? PA cos theta. Since it's perpendicular, we'll just take P into A, which means you have to differentiate A. Differentiation of A will give you A naught. Differentiation of 1 is 0. Differentiation of sine t cos. This EMF will go here and becomes a square. So when this EMF goes here and becomes a square, I will have my power directly proportional to cos square theta. So when it's directly proportional to cos square t or theta, it has to start from the peak. Those two are not the answers. So this will be the only answer for us. Cos square t is how it varies. Now let's go for the next chapter. And now we have a question from the last question which I'm going to discuss, thermodynamics. From thermodynamics, he has given a question from adiabatic expansion. He just gave you the difference in the temperature. So delta T is 10 and gamma value is 5 by 3. Asked us to find out work done by the gas. What is work done? Nr delta T by gamma minus 1. Actually, it has a minus sign. If you are taking delta T as T2 minus T1. So here, number of moles is 1, R value is 8.3 multiplied by 10 divided by 5 by 3 minus 1. How much does it give you? 83 divided by 2 by 3, which is multiplied by 3 divided by 2. 8 is 24,249 by 2 gives you 124.5 joules, which is option number 4. Thank you so much. Now we will go to the next questions and we have our sir to explain the review.
Hello guys, good evening everyone. I hope you had a great exam. So let us continue from the next part after what ma'am has discussed till now. Let us solve the next questions. So let's get started. I know you are eagerly waiting to know all the answers more than the process. So let us try to understand the process as well and then make sure you don't repeat the mistakes. So if you can see the first question, The position is given as a function of time. S of t is equal to alpha t square minus beta t plus gamma. So the question asks for average speed of the particle from t is equal to 0 to t is equal to 6 seconds. So what you are supposed to do, first differentiate this. So if you differentiate this, you will get it as, if you differentiate position, you will get velocity. So velocity of v of t becomes 2 alpha t plus, minus of gamma. So what you will do, alpha, beta, gamma values have been given. So first substitute that, that will give you S of t is equal to alpha t square means t square minus beta t means minus 60 plus gamma. Gamma value is given as 5. So if you differentiate this, you will get velocity, which will be 2t minus 6. Now the mistake students do here is they'll substitute it directly at 0 and 6 and they'll take the average velocity answer. But you have to check if the direction has been changed or not. When will the direction change? At the point of changing the direction, the velocity will become 0. So first equate it to 0. If you equate it to 0, you'll get the points where direction has been changed. So if you equate it, you will get t is equal to 6 by 2, so 3 seconds. So you have to first find displacement at 0 seconds, which will give you 60 plus 5. So if you put 0, you will get plus 5. And if you substitute 3, you will get 3 square, 9 plus 5, 14. 14 minus 6 3s are 18, so minus 4. So again substitute it as at 6 seconds. So that will give you, again you will get the same answer, 6 square minus 36, which is plus 5 again. So if you observe, it has traveled from plus 5 to minus 4 and again it came back from minus 4 to plus 5. So what is the total distance here? 9 meters this side and again 9 meters to the original position. The question asks for average speed and not average velocity. So total distance by total time gives you, total distance will be 9, total time is, total distance will be 9 plus 9 and total time is 6 seconds. So 18 by 6, option 3. 3 is your answer. So let's get to the next question. This is the easy question. The question asks for application of electromagnetic waves. What are the applications we use? We use microwaves for warming of food. UV rays are used for purifying of water. Radio waves, as you know, is the easiest. You can see this for FM and AM communication. And if you can see, the remaining option is 4. So answer is option 4. 2, 1, 4, 3. This is the very easy question. So let us get to the next question. A unit positive charge is taken slowly through an infinitesimally thin tube that is inside a charged dielectric sphere of radius r. So at first, students will feel terrified after seeing this question or the diagram, but if your foundations are strong, this is a very simple question. So observe here, there is a sphere, its radius is r, and there is a tube. So the charge has been moved from point A to point B. The question asks, what is the total work done? Okay, the total work done is also given, which is rho square by n epsilon naught. So the value of n is, you are supposed to find the value of n. So the point you have to understand here is, what is the data given here? Radius is r, uniform charge density rho. So as you know, density is equal to charge by volume. So you can get the total charge by multiplying density into volume. Volume of the sphere, as you know, is 4 by 3 pi r cube. So from this you will get the total charge. Now what is the total work done? Work done can be calculated as change in potential energy. So U final minus U initial. What is the electric potential energy formula? That is the core concept underlying here. As you can see, A and B are not within the sphere. They are outside of the sphere. So, and electric forces are conservative. So, it does not depend on the path and depends only on the final positions. So, you can simply substitute U final and U initial. What is the formula for that? U final will be KQ by R, U initial will be KQ by R2. And if you simplify it and equate it to the given value, you will get the answer as, you will get the required answer, which is 18. So that will be your answer. So understand that what we have done, first we have found out the charge using this formula, density is equal to charge by volume. Later we have used this concept of work done is equal to change in potential energy, and potential energy formula, if we are treating it as the point is outside of the sphere, this acts like a point object. So you will take the charge as q, you will substitute it, and in place of r1 you will keep 2r, in place of r2 you will keep 3r, and then equate it. That's how you will solve this problem. Let's see the next question. A current I flows through metallic circular loop of radius r. If you can see, this entire loop is made up of two parts. ABC has one type of resistance, ADC has another. What was the data given there? ABC resistance is half that of ADC. That means if this is R, its resistance will become R by 2. So if the resistances are different, will the current be same? Of course not. So the current through one wire will be I naught by 3. Using current divider rule, you can find it out. The current through the other part will be 2 I naught by 3. Now you know the current flowing through it, and can you see they are in parallel combination. From the parallel combination, you will apply current dividing rule, and that's how you will get the currents I naught by 3 and 2 I naught by 3. Now, are the because of a circular coil, current carrying circular coil, what is the magnetic field? You know the formula. B is equal to mu naught I theta by 4 pi r. As you can see here, theta is, this is a semicircle. So you will take theta as pi, that will make it mu naught I by 4r. Similarly, you will substitute this also, the radius is same, and if you simplify them, you will get the answer. One mistake students attempt here is, they think they can add both the magnetic fields. But if you observe, current is going in this direction and this direction. Applying right hand rule, by applying right hand rule, you can see current is coming out due to this wire. And if you see this wire, you can see current is going in, applying right hand rule. So one magnetic field is going in and one magnetic field is coming out. So the net field comes out to be B1 minus B2. You will just take the magnitude, because whichever is bigger, just take it, and you will get the answer as mu naught I naught by 12r. So option 2 is correct. Let us see the next question. A bob of mass m. Okay, in this question, basically what the question says is, a bob is hanging, it is at rest initially, and another mass of same, another object of same mass, it has been given in the question. Mass is m, so this mass is also m, and it is coming with a speed of 10 meter per second, and it collides with it elastically. So if you recall this topic, in elastic collision with same masses, what will happen? In elastic collision with same masses, velocities just get exchanged. That means after this elastic collision, the velocity of A will be equal to 0 and the velocity of B will equal to 10 meter per second. So now what happens? Apply law of conservation of energy. The kinetic energy at the bottom, when after hitting, this bob goes up to a certain height, and at the maximum height, what will be the potential energy? It will be equal to mgh. So this kinetic energy gets entirely transformed into potential energy. So what you have to do? All you need to do is just equate kinetic energy and potential energy, which is half mv square is equal to mgh. Masses get cancelled. So we want height here. So h will be equal to v square by 2g. What is the velocity here? Because it's elastic collision, the entire velocity will be exchanged. So it will get 10 meter per second. So the answer will be 10 square into 2 into 10. So 10 by 2, which is equal to 5 meters. So this is a very simple question. If you understand the concepts properly, you can apply it directly. The answer is option 4. So let us see the next question. A particle of mass m. Okay, this question is based on friction, right? So its function is given, mu k of x is given as mu naught minus alpha x. So what you are supposed to do, mu k of L is also given, which is equal to 0. Just substitute it here. So that makes it mu naught minus alpha into L is equal to 0. That means L is equal to mu naught by alpha, or alpha is equal to mu naught by L. What are you supposed to do here? you have to find the total work done. The value has been given and you are supposed to find the value of n here. So work done, as you know, is the integral of force into displacement. As it's frictional force, you will take minus F. In the place of force, what you will take? Frictional force can be given as Fk is equal to mu k into n, which is mu k into mg. Just substitute it here. Mu k, in this place of mu k, you will substitute mu naught minus alpha x into mg into dx. So this is what you are supposed to do. From which point to which point will you integrate now? As it is given from work done from 0 to x is equal to 0 to x is equal to L. So integrate it from this point to this point. How will you supposed to do? Mg is constant, take it outside. So integral of remaining part. So mu naught dx will become mu naught x minus alpha x becomes alpha into x square by 2. And if you substitute the values here, which is 0 and L, you will get the answer. That will become mg into mu naught L minus alpha L square by 2. Here in the place of alpha, you can substitute mu naught by L. Here you can substitute mu naught by L. So mg into mu naught L minus, in place of alpha, just substitute mu naught by L into L square by 2. L and L get cancelled, and if you observe, it will, I'll simplify it here again. W is equal to mg into, can you take mu naught L as common? If you take mu naught L common, you will get 1 minus 1 by 2. So which is nothing but 1 minus half is half only. So mgl mu naught. If you observe, it has been given as n into mu naught into mgl. So what is the value of n? It is 1 by 2, which is our answer. So let us get to the next question. Three media PQ are given. If you observe, the refractive indexes have been mentioned. It is 1, it is 1.25 and it is 1.5. And if you are observing from the top, what was the height? It was h1. And if you are observing from this medium, the height was h2. The question asks to find h1 minus h2. So this question is from ray optics and it asks for apparent depth. So as you know, apparent depth formula is real depth by mu. So most of the students go wrong here if they don't understand the exact process and they just mug up the formula. Most of the students, what they'll do, they'll take mu here and they'll take either 1 or 1.5. But what is the exact formula? It is mu relative. So if you are observing from the top, the apparent depth will be equivalent to, as it is at the midpoint, this will be 2.5 centimeters. So the real depth is 2.5 and relative means 1.25 objects refractive index by observer's refractive index. So that will give you 1.2, this will be 2. Similarly, if you are observing from bottom, what will happen? You are observing apparent depth will be, again, this depth is also 2.5 because it is given that object O is at the center. So that will be equivalent to 2.5 divided by 1.25 divided by, here most of the students make mistake here, they'll take either 1 or they'll leave it blank, but here surroundings also matter. So relative refractive index you have to take, which will make it 1.5. So if you substitute this, you will get as 2 into 3 by 2, which is 3. So the question asks for h1 minus h2. So 3 minus 2 is 1. So 1 is your answer. So let us see the next. A car travels on a circular racetrack of radius 50 meters. Okay, this is a very easy question which is on the banking of roads concept. As you know, tan theta is equal to v square by rg. Just plug in the values directly, you will get the answer. What is the value of v here? 10 meter per second. So 10 square by, what is the radius? Radius is 50. G you can take it as 10. So if you simplify it, you will get 1 by 5. So theta will be equivalent to tan inverse of 1 by 5, which is option number 2. So this is how you can apply the simple direct formula from NCERT. This is a standard formula. So here a frictionless circular wire is given. Okay, two objects, 2 points are starting from A. One is going in this direction and another is going in this direction. and you are asked to find the total linear momentum of the system as a function of angle here, not as time. If it is time, it will be a flat line, but since it is an angle, here it is going in clockwise direction, it is going in anti-clockwise direction. And as you know, momentum is nothing but mass into velocity. Here, what will you take in place of velocity? V is equal to r omega. Radius is unit only, mass is also 1 unit is given. So P is nothing but omega here. But the coordinates will be cos theta and sin theta. The horizontal components get cancelled out, but the total vertical component will be added up. So which will remain in the form of sin theta. So if you observe the options by applying common sense, you can see it won't be a straight line. So 1 and 2 you can eliminate. And if you calculate it, you will get added components of cos theta. So as you can see, 3 is also not a proper cos graph or sine graph. So option 4 will be the answer. So let us see the next question. 3 PN diodes D1, D2, D3 are connected as shown. The width of the depletion regions was given, which was W1, W2, W3. If you know the concept, if a diode is in forward bias and if a diode is in reverse bias, width will increase. So observe here in this diagram, the positive is connected to positive, here also it is connected to positive. So D1 and D2 are forward biased. So D3 is reverse biased. So you can blindly say that D3 will have more width. So see the options. D3 corresponds to W3 here. W3 is equal to W1 wrong. W3 is lesser wrong. W3 is equal to W1 wrong. So W3 greater than W2. So by eliminating options also you can do, if you just know the basic idea that in reverse bias width of the depletion region will increase. Let us see one question. Thin lens combination has been given. This is from ray optics. All you need to do is just apply the lens formula. What is lens formula? 1 by f is equal to 1 by v minus 1 by u. And what values will you substitute? First you will take that minus 30 for the mirror 1 and you will take the focal length plus 10. You will get a value v. So that image will act as object for our second lens. So that you will take as u2 for the second mirror. Again you will plug in focal length minus 10 and all you need to do is just apply the same formula of lens formula and you will get the answer as 60 centimeter to the left of concave lens. Concave lens. This is concave lens. Don't get confused. So let us see one last question. A solid sphere of radius r and mass m is attached. Okay, here this is a question which is based on parallel axis theorem. This inertia is given, You are supposed to find the difference of Ia minus Ib. So first let us see how you will find the moment of inertia for this setup of Ia. So as you know, it's a solid sphere. It will be equal to, first you are supposed to do inertia of this exact single sphere and then you have to do parallel axis theorem to include the moment of inertia of this B. So I'll just write it as Ia self plus Ib shifted. So what will be the answer now? For a solid sphere, you know, it is 2 by 5 m r square. For B shifted, what you will do? You have to do parallel axis theorem which says I naught plus m r square. Here you have to make sure you are doing the noting the right terms like small m and small r plus m into r square. What is r here? The distance between the two, which is r plus r whole square. Similarly, you will get IB. What will be the value of IB? Same process you will do it in the reverse manner, and if you do it, you will get as 2 by 5 small mr square plus 2 by 5 capital mr square plus capital M into r plus r whole square. This question at first looks like a very tough or difficult question, but it's just an application of parallel axis theorem. So by doing this, you will get I and YB, and if you want to do the difference, this gets cancelled and this gets cancelled. What you are left with r plus r whole square into m minus m. So you see carefully, it's not IB minus IA, it's IA minus IB. So the answer will be option 3. So that completes the set of 15 questions. Now I hope you have done well and you have learned from your mistakes, and I think you'll crack and you'll get good marks. Now I want to welcome our sir, physics sir. He will continue with the next set of questions.
Thanks, sir.
Thank you, sir.
Okay. So, good evening, everyone. So how is the dose? I think low, right? Because lots of you were not expecting this thing, but in resonance already long back, we announced that okay, paper will definitely shifted towards the tougher side. With respect to the previous experience also, we have seen the same thing that whenever there was a re-NEET, they have kept the physics part definitely tough. Even a whole paper they have kept tough. So obviously we are expecting the same, but we announced that okay, the paper will not be exactly tough like okay the way they gave it last time when there was a re-NEET, but definitely the paper is tough relative to the normal paper, right? Shifted to the moderate side, or we can say that okay, it is shifted towards the tougher side. So don't worry, if you have scored less, then also you have a hope, you have expectation. So all right, thanks, sir. So I think you all can work on this thing. So just keep on counting your marks. I won't waste lots of your time. Those who are going to give the exam next year, for them obviously we'll do the analysis and we'll tell lots of things after this discussion. But as of now, I'm going to the discussion directly. So here the question is, consider an electron revolving in an excited state of hydrogen atom with velocity this. The radius of orbit is this thing. We have to find the value of x. The value of x is equal to, okay, basically x we have to find, right? So over here, like it is a question of Bohr atomic model, first thing, from atomic structure chapter, where electron is revolving in the nth orbit, right? So over here we can write k q1 q2 upon r square is equal to m v square upon r. So from here R is equal to K Q1 Q2 upon M into V square, K 9 into 10 raised to power 9, Q1 obviously one of the charge will be a proton at the nuclei and the other will be electron revolving the orbit. So 1.6 into 10 raised to power minus 19 whole square upon mass of electron I think they gave already. They asked you to take 9 into, actually it's 9.1 something, but we are taking 9 into 10 raised to power minus 31 into v square. So velocity square, and velocity is given this one, so 25.6 into 10 to the power 10. Okay, now after calculating you are getting the value this thing. Okay, all right, so that will be the answer. Now moving to the next question. The mean free path of molecules in an ideal gas is half that of the ideal gas B. Okay, the mean free path of A is half the mean free path of ideal gas B. The diameter of the spherical molecule of gas A is twice. So, first thing, lambda A is lambda A is half of lambda b, right guys? First thing that is given. Second thing, that diameter, the diameter of A is twice the diameter of B, 2 times diameter of B. Now lambda is proportional to 1 by, they are asking number of molecules, like number density of A and B. So N into D. Okay. So from here we can do basically D square. So from here, by using this formula, we are getting lambda by lambda B is equal to NB by Na d a square, and by putting the values ratios, because already we have these ratios, we are getting Na by Nb is equal to 1:2. So from here Na is equal to half of this one. Okay, done. Moving to the next question. Moving to the next question, a cylindrical cork is of uniform density floats in a liquid of density rho A. If the cork is depressed slightly and released, it oscillates harmonically with time period capital T. If the same cork floats in another liquid of density rho 2, then the similar oscillation has time period 2t. The value of rho 2 by rho 1 is. So guys, over here, like we have done these questions that okay, it is any cork or any wooden log is at equilibrium. Okay, now we used to balance the force. So here mg is equal to like this buoyancy force, dip force. After that we used to compress it, like we used to dip it by distance x a bit, and then in that case what used to happen, that it used to come to unstable equilibrium, sorry, stable equilibrium, but it used to get displaced from the equilibrium position. So in that case the extra volume delta v was used to come x into area, right? This was the extra volume dipped, which will give the buoyancy force extra in upper direction, because downward mg is balanced already, but now this extra, so F net was coming as density of liquid into volume x into A. From here, mass into acceleration, so mass equal to cross section area A into length of the cork or wooden log, whatever it is, into density of the cork into acceleration is equal to rho L x into A. From here we got the idea that okay, A will be inversely proportional to density of the cork. Okay, so from here, like they are asking time period, so this will be inversely proportional, means, and we know that okay, this thing is coming proportional to what? Omega square, means omega will be inversely proportional to under root rho of the cork. Okay, so from here, time period, so time period is inversely proportional basically to omega, right? So here acceleration is coming like this, so put this thing here, so t1 by t2 is equal to omega 1, sorry, omega 2 by omega 1, right? And over here, like omega we got, density of the corks are given, so density is inversely proportional, omega is inversely proportional, so time period will be directly proportional, so t1 by t2 is proportional to what? Under root rho cork 1 by rho cork 2. T1 by T2 is given as 1 by 2 is proportional to under root rho cork 1 by rho cork 2. From here, rho cork 1 by rho cork 2, 1 by 4. Okay. So rho cork 1 by rho cork 2. So rho cork 2 by rho cork 1, rho cork 2 by rho cork 1 will be giving you this value. Okay. So check this value. All right. Now, so I think you are getting this idea. This is like the, if lots of students in NEET generally they remember the result also. So if you remember the result, then you can go directly. Okay. Moving to the next question. For sound waves, if the number of nodes for the fifth harmonic of an open end pi is n. So see, for open end, okay, we have a formula that frequency is equal to n by 2l into under root t by mu in that case, but here it is a organ pipe, so v, right? So here n is equal to 5, and for closed end, okay, in the other case they are saying that okay, ninth harmonic of the same pipe with one end closed. So it is a question from sound waves, so organ pipe topic. So here 2n minus 1, like frequency is equal to 2n minus 1 by 4l into v. So from here 2n minus 1 is equal to, and not in m, you can take m. So 2m minus 1 is equal to 9, m is equal to 5. So n is to m or m is to n, whatever they are asking is 1. So this is the correct answer. Okay, so it was a formula based question, not very hi-fi thing. Okay, just a basics one. Now moving to the next question. Consider the following nuclear reaction. So uranium, it is just being divided into two parts like thorium and helium, like radioactivity they are showing. Now in that case, masses are given. So they're asking that, okay, what is the Q value? So Q value, all of us know that Q value is equal to delta M into C square. That's it. Okay. So you don't have to do anything extra. So delta M you have to use, that is the main problem, that you have to calculate. Okay. Apart from that, there was nothing like uranium is given, this and this is given. So just put it here, 238.050. So 238.05 minus this one 234.043 minus 4.003 into C square. So 931.5. They have given also 931.5 only. Mega electron volt. Okay. So this will come in mega electron volt. After calculating, convert it into kilo electron volt. So you will be getting answer of this one. Okay. Just calculate it, it is a calculation. But obviously in the exam, it becomes really tough because data are very nearer to each other, so you can't do approximation here, so you will get that answer. Okay, moving to the next question. So which of the following measurement required index correction? This was something which was very basic, but it was tough for us because generally we don't discuss this thing as a student. Teachers generally while teaching they discuss this topic, but after that it rarely comes in an exam in the like mock tests and all. So that's why generally we forget it. Okay, so those who remember, they have done it, this is very easily. That is it is a measurement of what? Focal length. Okay, so index correction is what? Like it is the measurement of focal length of lens using optical bench. Okay, so that is the thing what we have to remember. So basically those who have already given the exam, for them we don't have to do anything with this one, but those who have to give exam next year, for them it is useful, they should remember this thing. Okay, moving to the next question. In the solar system, so this was a very easy, right? In a solar system, time period of revolution of the planet tracing the circular orbit of radius r is proportional to, so t square, all of us know that is proportional to r cube, right? So this was very easy for us. So time period will be proportional to r raised to the power 3 by 2. So this is the answer. Okay. Now moving to the next question. Consider that rho s, Kb and B represents Stefan's Boltzmann constant and Wien's displacement law constant respectively in the dimension of this thing. So guys, here there is nothing to explain. If you know the formula, you can easily, because you know that how to calculate it. So first thing, that Stephen Boltzmann constant. So it is given as energy falling or radiated per unit time. Energy radiated by any body having temperature T per unit time, that is power. So power is equal to sigma E8 t raised to the power 4. Power is m l t to the power, sorry, ml square t to the power minus 3, right? Energy by time. So ml square to the power minus 3 equal to sigma E, don't have any dimension, dimensionless quantity, l square and k to the power 4. From here I will get sigma. Okay, easily. Okay, all right. After that, next is Kb. So over here, Boltzmann constant. So Boltzmann constant, joule per Kelvin, because like Kt, in that form you can have energy equal to Kt. So from there, Boltzmann constant equal to joule per Kelvin, like this you can use. So that is the thing. And coming to Wien's displacement, so lambda equal to b by t. So b equal to lambda into t. Lambda wavelength. So dimension will be a into Kelvin. So just after calculating all these things, I think you will be getting this one. But anyhow, that is a normal calculation. Just multiply all of them, you'll get the answer. So it was a very basic question, but the dimensions which were given was bit calculating. Now, going to the next question, a ray of light with wavelength lambda is incident on three different photoelectric cells, namely this. Okay. So, 1, 2, 3 photoelectric cells are lambda 1, lambda 2 and lambda 3 respectively, and the magnitude of stopping potentials are V1, V2 and V3 respectively. The relation between lambda and the threshold wavelength. So, see guys, lambda 1 is less than lambda. Okay. So V1 will be 0, either this option or this option, right? But in the other, on contrary here, lambda 2 and lambda 3 is greater, but lambda 3 is very greater than what? Lambda. So obviously V2 will be less than V3 because lambda 3 is greater. So obviously this will be the answer. Okay. So logically here, you don't need to solve it completely, just logically we can get this answer. Now moving to the next question. One of the scale. Now see, here this is a question from vernier caliper. Here one of the scale of the vernier caliper is equal to 1 millimeter and the number of division on the vernier scale is 10. Means this is a normal vernier caliper scale. Now when both the jaws touch each other, the vernier scale shifts to the left of the zero. So of the main scale, and the way fourth vernier scale division coincides with the division of the main. So see, it is a case of negative error, right? Already means when it is showing 1 millimeter, if the, like if you are measuring something with that scale, and if it is saying showing 1 millimeter, means actually its real length is more, right? Because to make it 0, first you will have to shift it on the other side. So here this is showing negative 0 error. So in that case, already the answer will be, if the length of the wire shown be 1 centimeter, so obviously it will be more than 1, that is 1.04. So no need to calculate it. Even if you are calculating, then also the 0 error is least count, that is 0.1 millimeter into the division. So fourth division of the vertical capital. So 0.4 millimeter or 0.04 centimeter. So you will add with 1, then also you will get the same thing. But no need to waste the time like this because other options are not given like that. Okay. Now coming to the next question. Here, like a point charge is placed at the center, right? Inside the solid conducting sphere. Now this is important. Conducting sphere is there. So what will happen? Here minus q charge will come, which will be here, and the same plus q charge will come here. So this will act like this. Due to this one, electric field over here or here will be 0 due to the inner system, because flux will be 0. So this will act as a point charge kept at its own center. So first thing, that here EA is not equal to 0. Inside it, electric field will not be 0 because charge is there at the center. But apart from that, EB will be equal to EC. So this is the, because this will act as a sphere kept at its own center. Now moving to the next question. In Marsden experiment, the number of scattered alpha particles n theta is plotted as a function of scattering angle theta. Now this is something which is experimental, and this is given in your NCERT also directly. You have to remember this one because n theta will be proportional to sine to the power 4 theta or proportional to 1 by theta to the power 4 if the theta is very small. In that case, it's something like this. Okay, so graph will be like this. Moving to the next question. One mole of an ideal monoatomic gas undergoes a cyclic process as shown in figure. The total heat supplied. So here, as it is a cyclic process, so total heat supplied will be what? Equal to work done, because delta U will be 0. Delta U is 0. So delta Q is equal to delta U plus delta W. So find delta W. That's it. Done. Okay. Delta W what? That is the area of this. So area is, this is already Newton per meter square and this is meter cube. So all of them are given in the SI unit. So this is 200 and this is 3. So 600. Okay. Now moving to the next question. two infinite long parallel conducting wire A and B carry currents I and 2I respectively in the same direction. Wire A has uniform mass density lambda and lies on the insulating floor. So this is the ground on which a wire is lying here. Okay, on the floor. The B wire is kept at a height h above the floor. The minimum magnitude of h. There is another wire B which is at a height h above the floor. So minimum magnitude of h, so that the wire A does not rise on the floor. So this is the question from magnetism, where we are applying a concept of force between two parallel wires, current carrying wires. So here they are carrying a current. So downward mg will be acting. The force in upper direction will be F is equal to mu naught I1 I2 by 2 pi d into L. So here equate it. So mu naught I square by 2 pi d is h is equal to mg. M is lambda into L into G, and for length L this will be L. So L will get cancelled. So from here we can get H equal to mu naught I square by 2 pi lambda G. Okay. So mu naught I square 2 pi lambda G. That is the correct answer. Okay. Now moving to the next question. Consider a spring mass system harmonic oscillator in one dimension. The mass of the particle is m kg and the spring constant is k Newton per meter. At a given instant, the extension of the spring is x meter. Alright? And the particle speed of the particle is v meter per second. On the x versus v plane, if the graph v as a function of x is a circle. Now this is very important to remember that this is a circle. So this is a question from SHM in which they are using a concept of spring mass system as well as the graph between v as well as x. Okay. So position versus v graph, we all have learned that that will be elliptical, but it becomes elliptical only if omega is not equal to 1. So they change the whole concept what we have learned in normal classes. In normal class, like we remember that v square equal to omega square into a square minus x square, right? So from here, it becomes the equation of circle only if this omega becomes equal to 1, isn't it? Only in that case, v square plus x square is equal to a square, means y square plus x square is equal to r square. So this is a mathematical equation of circle. We will not go in such detail, but if this has to become a circle then. So this was bit tough for us because we are not related much with the like mathematical tools. But anyhow, if you can remember this thing that okay, this is the equation of ellipse and then we can convert it like this, then in that case omega is equal to 1. Now in case of spring mass system, omega 2 pi under root m by k, from there we know that okay, omega is k by m is equal to 1. Squaring both sides, k is equal to m. So this is the okay, done. So that's it for physics. I think you all got the idea that okay, it was shifted towards the surface side and we were having that idea. Anyhow, now it's a time for the last part of discussion, that is chemistry. So now we'll start the chemistry. Vikas sir, welcome. Okay, thanks.
Thank you, sir. So let's see the first set of questions from chemistry part. And overall chemistry, if you see from the May 3rd paper, it is more logical, especially in physical and organic chemistry. Questions were made logical. And in inorganic chemistry, more weightage is there and more simpler questions are there that the students can score in this inorganic chemistry part. So now let's see the questions from the organic chemistry part. First question, if you see, consider the following reaction and choose the correct option. Toluene is given and toluene is treated with a CrO2Cl2, chromyl chloride, in presence of carbon disulfide solvent and followed by hydrolysis, protonation you can say, right? So in this one, This is the question coming from the aldehydes, ketones and carboxylic acids. So what is the point he has given? Toluene is treated with chromyl chloride. So chromyl chloride, we know it is a oxidizing agent which will oxidize the terminal methyl group into the aldehyde. So you will be getting here the compound P as this one. Benzaldehyde you will be getting. And now this is the compound P. Now let's say choose the correct option, right? Compound P is obtained by the hydrogenation of benzoyl chloride with palladium and barium sulfate, right? So do we get this compound when we reduce benzoyl chloride? Benzoyl chloride is this one with palladium barium sulfate. Yes, correct, right? So this is correct. On treating compound P with a saturated NaHCO3 solution, brisk effervescence is observed. This is wrong because the compound we obtained is aldehyde and this is the point related to carboxylic acids. So this is wrong. Then compound P can be prepared by treating benzene with anhydrous AlCl3 and acetyl chloride. No, we don't get benzaldehyde, we will be getting acetophenone with this one. Wrong. On treatment with bromine water, compound P gives a white precipitate. So when this compound is treated with bromine water, we don't get white precipitate here. Right now? So this is also wrong. This is also wrong. So then going to next question, the compound that cannot be obtained from aldol condensation. So it is also from the same chapter, right? Little more carefully we have to solve it, right? First of all, this is a compound, two organic compound, carbonyl compounds treated with NaOH. So it is a crossed aldol condensation reaction, and in this crossed aldol condensation reaction, only this carbonyl compound is having alpha hydrogens. This is not having alpha hydrogens. So one having alpha hydrogen, one not having alpha hydrogen. There are two possibilities are there. That is a crossed aldol product to be formed between these two, and one simple aldol product will be formed between molecules of the same compound, two molecules of the same compound. So if I take this one, double bond O, CH3 plus, or I will take it like this. In a similar way I will do, okay na? Double bond O. These are the alpha hydrogens, right? Carbonyl group, adjacent carbon is alpha plus benzaldehyde. CH, CH double bond O. So if there is elimination of water molecule, you will be getting the crossed aldol product as this one. But understand one point here. This is the carbon-carbon double bond where geometrical isomerism is possible. Both E and Z can be formed here. That is the reason why here he has given both the products. So both will be formed as a geometrical mixture. And coming to simple product. Simple product is when this compound is reacting with one more molecule of the same compound, that is right. So here dehydration will happen. Aldol condensation is overall it is addition plus elimination. Together it is condensation, and there dehydration is the one which will happen. So if this water molecule is eliminated, what you will be getting here? So that is this compound. So what is that compound here? That is this one. So this also will be formed. So what is the compound which will not be formed is option 2. So he is asking for the incorrect answer. So this is option 2 is incorrect here. 1, 3 and 4 are correct, right? So little more time it will take, but question can be done. There is no problem in it. For the following reaction sequence, choose the correct option. There is no reaction sequence given here. I have the question paper. I will write it here as it is missing, right? for the following reaction sequence. For the following reaction sequence, choose the correct option. Actually, question is given it as, reaction is given as benzene treated with a CH3, C double bond O, Cl in presence of AlCl3. And as a second reagent, he has given it as NaOCl, sodium hypochlorite, right? So he is asking, this is a P plus Q, and he is asking the question about P and Q, right? Now first let us solve the question. Benzene on Friedel-Crafts acylation. What do we get? Benzene on Friedel-Crafts acylation. With the first reagent, we will be getting the product as acetophenone. Now this acetophenone you are treating with a sodium hypohalite. So it will be undergoing haloform reaction. During the haloform reaction, this will be broken, that you will be getting from with the second reagent, you will be getting that plus chloroform. Sodium benzoate plus chloroform will be getting. So sodium benzoate is P, let us assume, then chloroform will be Q. Now let's come back to the statements here. Both P and Q are carbonyl compounds. Is it both P and Q are carbonyl compounds? Both are not carbonyl compounds. So this is wrong. This is wrong statement, right? Second, if P is the sodium salt of a carboxylic acid, correct? Q is a primary alcohol, wrong. So overall, this is wrong. This is also wrong. P and Q are aromatic compounds. No, only P is aromatic compound. Q is not aromatic compound. This is also wrong. Only leftover option is option D. If P gives a carboxylic acid on acidification, if you do acidification, definitely you will be getting benzoic acid. And Q gives a poisonous gas on exposure to air and light. Yes, obviously, this on exposure to air and light, this will get converted into phosgene, COCl2 phosgene, right? So this is a radical reaction. So this statement, both points are correct here, that fourth option will become the perfect answer. Now going to the next question, one of the products formed in the following reaction is, first this is a Grignard reagent, right? Grignard reagent is what it is, R minus MgBr plus. Now this R minus, whatever the strong base is there, that will abstract this acidic hydrogen. So that results in the formation of R minus with H plus RH. So this is R minus, which will take the H plus. So you will be getting cyclohexane plus NH minus MgBr plus. These are the two products we will be getting. So one of the products formed in the following reaction, benzene is given in the, cyclohexane is given in the first option. So that itself is the answer. Now going to the next question. Given below are two statements. Statement 1, oxidation of para-nitrotoluene with acidic camphor. Acidic camphor is a strong oxidizing agent. Therefore, what will be the product formed here? When para-nitrotoluene is treated with a camphor, we will be getting para-nitrobenzoic acid. Para-nitrobenzoic acid, right, gives an acid that is stronger than benzoic acid. Obviously, NO2 will be increasing the acidic strength, so it is stronger than benzoic acid. Statement 1 is correct. Statement 2, reduction of para-nitrotoluene with the Sn plus HCl. So what will happen? NO2 will be reduced to NH2. So we will be getting CH3NH2. followed by neutralization gives an amine. Yes, it is an amine that is more basic than aniline. That is also correct because methyl group is an electron releasing group. It will increase the basic character, right? So statement 2 is also correct. So statement 1 is incorrect, but statement 2 is correct. Both statement 1 and 2 are correct. Option 2 is the answer, right? Simple only question. Now, identify the reactions which give aniline as the major product. Which gives aniline as the major product. First of all, if benzene carbonitride is reduced by lithium aluminum hydride, what do we get as a product? We will be getting not aniline but benzylamine. So this is not the compound, right? Then B, benzamide, right? This benzamide, when it is treated with KOH plus Br2, the C double bond will be removed. Of that, you will be getting Hoffman bromamide reaction, right now? So you will be getting aniline. So B is correct. Coming to C, nitrobenzene reduction with sodium borohydrate, no reaction, right? And this compound, whatever amide is there, R C double bond O NH PH, whatever is there, treated with HCl followed by hydrolysis. So this is acidic hydrolysis, that it will be broken here and you will be getting aniline plus acetic acid. So here also aniline is formed. So in both B and D options, aniline is formed. So this option 3 is the correct answer, right now? Next question. The correct statement about peptides and proteins is, in alpha helices, the polypeptide chain is twisted into a left-handed. No, it is right-handed. So wrong. Tertiary structure of proteins has 2 or more polypeptide chain. No, tertiary structure will be having single polypeptide chain. Tertiary structure. Quaternary structure is the one in which 2 or more polypeptide chains can be there, but that is also not mandatory, right now? So 2 is also wrong. 3, only the proteins having a quaternary structure are biologically active. That is also wrong, because even there are many proteins in which they are in tertiary structure and still biologically active. So only 4th option will be correct, in which in beta plated sheets the peptide chains are held together by intermolecular hydrogen bonding. Perfect, right? From biomolecule chapter. Then going to the next question, the amino acid. This question is also again from biomolecules. The amino acid that gives a red blood color on treating with the sodium fusion extract with the sodium nitroprusside. The compound which gives red blood color with sodium fusion extract and sodium nitroprusside is the compound which has sulfur. So who is the amino acid which is having sulfur atom in it? Not serine, in which oxygen will be there. Then leucine, no sulfur. Threonine, no sulfur. Methionine is the one in which sulfur will be present. Along with the methionine, cysteine is also another amino acid in which also sulfur will be there. So that sulfur with the sodium will be forming sodium sulfide, and that sodium sulfide with the sodium nitroprusside will be giving red blood coloration, even sometimes violet coloration also, right? Then next question, match the vitamins in list 1 with their sources. This is again from biomolecules chapter. So vitamin A will be having a good source of carrot. In childhood, your mother says, right, eyesight has to be better, eat more and more carrot, like that. So that is at this point. Then vitamin B12 will be present in meat. Then vitamin E will be present in sunflower oil, and vitamin K will be present in green leafy vegetables. So it will be A4, right? A4, then B1. This is the answer. Option 3 will be the answer. Then going to the next question, the following carbocation is stabilized by the interaction of the empty p orbital of carbocation with, right? And so this is the question from the GOC part, that if you take it here, CH, empty p orbital is there with CH2H. This is a sigma bond. This is a pi bond, right? So filled sigma bond and filled pi bond. This is a filled sigma bond, this is a filled pi bond, right? Completed. Next, arrange the following compounds in the increasing order of polarity. We know carboxylic acid is of the highest polarity. After that, you can take the alcohol and then acetone and then ether. So the highest from lower to higher we are going, right? The least will be A, then second least will be C, then better will be B, and best will be D. So this is the order of polarity A, C, B, D. Where is it? A, C, B, D, A, C, B, D. So first option itself is correct. Going to the next question. This question is important. See, more of a difficult also I can say. And this question is again from hydrocarbons reaction. Trans 2-butene upon treatment with bromine in CCl4. What is the trans 2-butene? Trans 2-butene is this one. Bromine. Right. So this is trans compound and it is anti-addition. Trans compound with anti-addition. What should we get? We should get a meso product. Right. Meso product we should get. That is MEH Br if you take, then MEH Br. This is a meso product. But the compound given is not a meso compound. This is optically active compound. Optically active compound. You bring methyl here. So two methyls will be coming closer. So then if this methyl is brought here, this hydrogen will go. So bromine and hydrogen are not matching. Right now? So you don't get the meso product here. So statement 1 is wrong. Then coming to statement 2, cis-2-butene on treatment with alkaline, cold alkaline dilute KMnO4. So here you will be getting cis-2-butene with cold dilute alkaline KMnO4, that is the Bayer's reagent. You should get again meso compound because cis compound with syn addition. Syn addition, meso product. So that is MEHOH, then again MEHOH. And that is the meso product given here. If you bring this methyl here, methyl overlapping, then this OH will be overlapping. This HH also will be overlapping. Right now? Then, among the following, the compound having conjugated double bonds is conjugated double bond in a hepta, that is a 3, 4, 5, 6, 7. Right? 1, 2, 3, 4, 5, 6, 7. Conjugated means here to be double bond and alternately here to be double bond 1 and 3. That is option 2 from hydrocarbons question. Then consider the following reaction sequences and choose the correct option. The compound given is this one, and to this compound what he has done, treated with Lindlar's reagent, and this is for syn addition. Syn addition. So for syn addition, what do we get here? We will be getting PHCH double bond C MEH. This is compound K. Now this compound K is treated with HBr. So when you treat it with HBr, what do we get here? It will be Markovnikov of addition will happen, that you will be getting it as a PH CHBr CH2Me. This will be the product M. Right. Now this side let us come. When this alkyne is treated with a Birch reduction, you will be getting trans addition. So C double bond C PHH HME. Right. So this is a compound L. Now this compound L, when it is treated with HBr in presence of benzoyl peroxide, it will be undergoing anti-Markovnikov addition with formation of more stable radical. That is, you will be getting PHCH2 CHBrMe. So this is the compound N. Right now. So if you compare here the statements, M and N are stereoisomers. This is N, this is M. But actually M and N are positional isomers here. So this is wrong. K and L are geometrical isomers. This is L trans isomer and this is K cis isomer. So this is correct. Understanding a simple question, but it will little take more time in writing the correct products because stereochemistry is a very important part here. You have to take care. So now the remaining questions will be taken care by Saurabh sir. Please, sir.
Thank you, everyone. Hello, my dear students. I hope you all get your NEET exam very well. I will discuss your physical chemistry questions. Now, what my view on the physical chemistry question? Talking about the physical chemistry question, it's a model. Some calculations are there, some tricky calculations are there. One by one, we'll go through all those particular questions. See this first particular question. Question says 2 moles of an ideal gas undergo free expansion from 10 liter to 100 liter at 300 Kelvin. The values of delta system and delta surrounding. We need to calculate entropy of system and surrounding. In this particular question, they mentioned free expansion. We know for free expansion, the work done is our 0. Now, in this particular case, whenever we are going from the volume expansion from 10 liter to 100 liter, the temperature remains constant, and we know when temperature remains constant, our internal energy is also 0. Now, first we calculate the entropy of a system. Now, in order to calculate the entropy of a system, we know it is our NCV ln T2 by T1 plus we have is our NR 2.303 NR log of V2 by V1. Since temperature is constant, so this term is our 0. So our entropy of system is equals to 2.303. Since in this particular question 2 moles of an ideal gas is given, so it is our 2 multiply R log of V2 by V1. Final volume is given as 100 liter, and in this particular case initial volume is our 10 liter, log 10 which is 1. So entropy of system we get is our 4.606 R, that is the entropy of system. Now in this particular case, one more thing we need to calculate the entropy of surrounding. We know our entropy of surrounding is equals to, our entropy of surrounding is equals to negative of heat of our system divided by temperature. Since in this particular case our work done is 0 and our internal energy is 0, so from our first law of thermodynamics our Q is also equals to 0. So from that, if our Q of system is equals to 0, that means our entropy of surrounding is equals to 0. So entropy of system is equals to 4.606 R and our entropy of surrounding is 0. That means the first option is the right answer. This is the right answer for this particular question, our option 1. Because in other options, in this particular case, entropy of system and surrounding both are 0, so wrong option. Similarly, in case of our fourth option also. Next question, moving to the next particular question. Again, thermodynamics question is there. Question says, consider the reversible process 1 mole of an ideal gas as shown in the figure. Work done 2, work done 3 and work done 4 are representing our work in calories. This particular 4 process is given. Here the important part is our second and fourth particular process is our adiabatic. And we know one thing, that for an adiabatic process our heat is 0. Now if our Q is 0, we know one thing, that our according to internal energy and our first law of thermodynamics, our delta U is equals to Q plus W. If our heat is 0, that means our work is equals to delta U. That means since for the process second and for the process 4 it is adiabatic, that means work done for second process is equals to delta U2 and work done for 4 process is equals to delta U4. That means our W2 plus W4 is delta U plus delta U4. So this is the most confusing option. That's why this is not the right answer. I hope so many students did the mistake. Now in this two particular case, now moving to the work W1 and W3. Now in case of your 1, since temperature is constant, that means it is an isothermal process, and we know one thing, that work done for an isothermal process is it is equals to minus 2.303 nRT log of our V2 upon V1. Now in this particular case, simply in terms of ln, if I write R in the question, they have given 2 calories per mole, so we'll get our minus 2T1. In terms of ln, it is V2 by V1, so that is our W1. And talking about the W3, our W3 is also the same case, we can write it as minus of, again it is our 2, but in this particular case our temperature is T2, and that is our ln of V4 upon V3. That means our W1 plus our W3 is equals to minus 2T1 ln of V2 upon V1 minus 2T2 ln of V4 upon V3. That means for this particular question, second option is the right answer. These two particular options somewhat tricky is there, but you need to be very careful in the concept. Second option is our right answer. Now moving to the next particular question. This particular question says, a protein undergoes reversible thermal denaturation from its initial state N to a denatured state D. So equilibrium is given between N and D. At 60 degrees Celsius, the concentration of N and D are equal. Means in the question they have given, the concentration of N and concentration of D are equal, and the standard enthalpy change of denaturation is also given. We need to calculate the standard entropy change at our 60 degrees Celsius. Now, first of all, if I write the K equilibrium for this particular case, we know K equilibrium is our product concentration and divided by our reactant concentration. So product concentration, that is our D upon N. Since these two particular concentrations are equal, that means my equilibrium constant is equals to 1. Now we can calculate our Gibbs free energy from equilibrium constant. We know that our delta G is equals to minus RT ln K. Now from this particular equation, since our ln K is K equilibrium is our 1 and we know log 1 is our 0, that means in this particular question our delta G is 0. Now apply our Gibbs-Helmholtz equation. We know our delta G is equals to delta H minus T delta S. Now because delta G is our 0, so we can write our delta H minus T delta S is equals to 0. So delta H is equals to T delta S in this particular question. Now enthalpy is given in the question is 666 kilojoule. So that is our 666 kilojoule per mole. Very cautious in the SI units what is given in the question and what they are asking. Now in this particular question, temperature they have given is a 60 degrees Celsius. We know temperature in degree Celsius is a 60. Now temperature in Kelvin is our 60 plus our 273, that is our 333 Kelvin. Now temperature is a 333 Kelvin and delta S. So standard entropy change in this question is our 2 kilojoule per mole. So our right answer is a 2 kilojoule per mole. That is our second option, 2 kilojoule per mole, because in the question they are asking kilojoule per Kelvin per mole. So that is the right answer, which is the second option. Now moving to the next particular question. For an elementary chemical reaction, the Arrhenius plot is given below. If the energy of the activation in the question is given as 6.64 kilojoule, R is also given, the temperature at which our K becomes E square per minute. This is what they have given in the question. Now simply this is an Arrhenius equation question, and we know according to the Arrhenius equation, our ln K is equals to ln A minus EA by RT, and this is a plot between ln K versus 1 by T. Now in this particular equation, we know that our ln A, this is our y-intercept, because if I write the equation y equals to mx plus t, so my y-intercept is ln A, and from the graph clearly I can see my y-intercept is 6. That means ln A in this particular question is a 6. Now they are asking calculate the temperature where your rate constant is E square. That means my K is E square. So I can write ln E square, that is equals to 2 ln E, and log base is equals to 1. Now we got the value of ln K, that is 2. And we got the value of ln A, that is also 6. Now only one thing, just put everything in this two particular equation. Now our ln K, that is our 2 equals to 6 minus EA upon RT. So simply I can write my EA upon RT is equals to 4. So temperature is equals to, activation energy is given in the question 6.64 kilojoule, 6.64 kilojoule per mole converted into joule. Now R is given in the question 8.3 and 4. After solving this mathematical equation, we'll get our temperature 200 Kelvin. So the right answer for this question is 200 Kelvin. I hope you all understand this question. Now moving to the next particular question. Next question says 2A gives B is a zero order reaction where K is given in the question, and if the initial concentration of A is 2 molar, then what is the time taken to complete 75% of the reaction? Now usually whenever a zero order reaction is given, we use our A equals to A naught minus KT. But in this particular question there is a twist, because here they have given for 2 moles. So simply if I apply minus 1 by 2 dA by dt, for a zero order reaction we simply write our K. So minus dA by dt is equals to 2K. When we will integrate this particular equation, we will get our A equals to A naught minus 2 kT. That is the main particular point in this particular question. Now, concentration initial is given as 2 and the final concentration at T when 75% of the reaction completed. So when 75% of the reaction completed, so concentration at time T will be, so 75% if we calculate for our 2, so it is equals to 1.5. So that means how much is the remaining at time t? That is a 0.5. So simply put the everything in the question, 2 minus 2 kt. K in the question is given as 1, so minus 2t. So our 2t is equals to 1.5. So our time is equals to 1.5 by 2. That is equals to 0.75 minutes. So the right answer is third option, which is 0.75 minutes. I hope you all understand this particular question. Now moving to the next question. This question says, this question is from electrochemistry. Question says, the standard electrode potential for the half cell, this equation we need to calculate at 298 Kelvin from the given data. Now first of all, we know directly we can't use in this particular equation our inert cell. First of all, we need to calculate our Gibbs free energy. Now, for this particular equation, Fe3 plus electron gives us Fe2 plus. So, our delta G is equals to minus NFE in this particular equation, and number of electrons gain is our 1. So, minus Fe. Now, what is given in the question? First given in the question, Fe3 plus will Fe. So for this particular case, 3 electrons it will gain. So our delta G for this equation is minus 3F, and for that particular equation, inert cell is minus 0.04. So that is our minus 0.04. So that is equals to 0.12 Faraday. Now delta G for the second equation, Fe2 plus gives Fe. So Fe2 plus gives Fe. Here the gain of your 2 electrons is there. Again we'll do the same thing. Delta G for this equation is minus 2F, and for that equation inert cell is minus 0.44. So that is equals to 0.88 Faraday. Now this is my target equation. In target equation, Fe3 plus on the reactant side. So simply what I need to do, from this particular equation I need to subtract this equation. So after subtracting this equation, if this is my 1 and if this is my 2, subtract 2 from 1. When we'll do this operation, subtract 2 from 1, so we'll get our 0.12 Faraday minus 0.88 Faraday, that is equals to minus 0.76 Faraday, and that is equals to minus Fe. Minus minus cancel, F is cancel. So E naught cell for the equation is 0.76. That is our third option is the right answer, 0.76. Now moving to the next particular question. Next question says, for a salt XY which is a strong electrolyte, the plot of lambda m versus root C has a slope of minus 90. At 0.01 molar concentration of XY, we need to, the value of lambda m is also given. We need to calculate the limiting molar conductivity of Y ion. Now we know first of all, according to our Debye-Huckel-Sagar equation, our lambda m is equals to lambda m naught minus A root C. Now in this particular case, my slope is minus A, and that minus A slope they have given in the question as minus 90. So from that the A value is our 90 Simon centimeter square, mole minus 3 by 2, liter 1 by 2, the unit is given in the question. Now concentration is given 0.01 and molar conductivity is also given, that is 145 Simon centimeter square per mole. Now if I put everything in this particular equation, our lambda m is given 145, that is equals to lambda m naught minus A root C, that is our 90, and root C is our root of 0.01, which is our 0.1. So we'll get here 145, that is equals to lambda m naught minus 9. So lambda m naught is equals to 154. Now lambda m naught, limiting molar conductivity, using with the help of Kohlrausch law we can write molar conductivity of X plus and molar conductivity of Y minus. Now this we have calculated 154. In the question they have given for our X plus is a 74 Simon centimeter square, that is a 74. plus molar conductivity of Y minus. So molar conductivity of Y minus is 154 minus 74, that is equals to 80. So our right answer is 80, that is a limiting molar conductivity of Y minus. Now moving to the next particular question. Next question says the correct order of the solubility of the given salt in water. Usually we know when Ksp is high, our solubility is high. But that is applicable when the number of ions they are releasing in the solution is the same. But in this particular case, the number of ions they will give in the solution is different. So first we will calculate their solubility value. How we will calculate? Simply for AgBr, we can write that is equals to Ag plus and that is our Br minus. So for that our solubility is Ss. So simply we can write our Ksp is equals to S square. So in this particular case solubility of AgBr is equals to root of Ksp, and Ksp for AgBr is given 5 into 10 power minus 13, which we will get approximately 7 into 10 power minus 7. Now for second case, that is our ZnOH twice. How ZnOH twice will give? It will give our zinc 2 plus plus 2OH minus. Now for Zn2 plus the solubility is S, and for OH minus it is 2S. So simply we will find in this particular case our Ksp is S multiplied 2S whole square, that is our S cube is equals to Ksp is given 10 raised to the power minus 15 divided by 4, and our solubility is the cube root of this particular value, which we will get approximately 6.8 into 10 raised to the power minus 6. And the last case is our Hg2Cl2, that is equals to our Hg22 plus 2Cl minus. Again this is S and this is our 2S. Simply again we can write our S is equals to, the Asp value is 1.3 into 10 raised to the power minus 18. This again is equals to our 2s square multiply s. So same thing will be here, again divided by 4 and do the cube root. Again you get the value after solving 6.8 into 10 raised to the power minus 7. So if I compare the S value, the maximum S value is for the zinc OH twice. So that means the solubility ZnOH twice is the maximum. Now moving to the next point, next wave is our AgBr and third is our Hg2Cl2. So the right order is our ZnOH twice is greater than AgBr is greater than Hg2Cl2. Now moving to the next particular question. Next particular question says the number, they have given the two numbers and we need to calculate up to 3 figures decimal point. First of all, we know one thing, whenever the last digit is 5, in that particular case, if the preceding digit is our even, in that case we should not change, as it is we need to write. Just like in the first number we have a 17.0145. Now we need to round off, but the last digit is 5, but here the preceding digit is even, so in that particular case no change. So simply after round off we will get 17.014, but in the next case we have is our 21.0235. Here the preceding digit is odd, so we need to round off to the next even number, and the next even number is our 4. So we will get 21.024. So first case is our 17.014 and the second is our 21.024. So fourth option is the right answer in this particular case. Now moving to the next question. Next we have is our amount of carbon dioxide evolved on complete combustion of 116 gram of N-butane. So first of all, what is N-butane? N-butane we know is our C4H10. It undergoes combustion to give carbon dioxide plus water. And how much gram is given? 116 gram is given. We know for the butane, its atomic mass is, because in this case we'll calculate the molecular mass. The molecular mass of our butane is, for butane the molecular mass is 48 plus 10, that is our 58. So for that our moles is equals to, in this particular case is 2 moles. So simply we can apply our POAC, principle of atomic conservation, on carbon. So if we say here carbon is conserved, so in this particular case we have 4 multiply mole of carbon because in this particular case 4 carbons are there. So 4 into mole of carbon is equals to carbon dioxide. 1 carbon is here, 1 into mole of carbon dioxide. So in this particular case 4 into mole of carbon in your N-butane. This is our butane. In our butane it is 2 moles. So our mole of carbon dioxide is 8, and we need to calculate the mass. So mass of carbon dioxide is 44 multiplied by 8. So we get 352 gram. So right answer for this question is 352 gram. Simple question is here. Directly we can do our POAC on carbon. Now moving to the next particular question. Next question says, consider the following statement about the solution formed by mixing two liquids. So these particular three statements are given. An ideal solution thus formed obeys Raoult's law throughout the composition range. We know an ideal solution obeys our Raoult's law. That means our first statement is correct. Now second statement, mixture of chloroform and acetone shows the negative deviation. That statement is also correct because we know the interaction is stronger. That's why chlorine and acetone, they shows the negative deviation. And in the third case, the mixture of aniline and phenol, it shows positive deviation. This statement is wrong because aniline and phenol, they show the negative deviation. So the right answer for this question is our A and B only. Now moving to the next question. Next question is, for an ideal solution formed by mixing P and Q, the delta H mixing is a 0, delta V mixing is a 0. This statement is correct because we know what is an ideal solution. For an ideal solution, the PP interaction and QQ interaction, this is the normal pure liquid interaction, and when after mixing, the interaction is the same in our PQ. Such type of solution is an ideal solution. Reason says no interaction occur between P and Q. This reason is wrong because interactions are there. So that means assertion is true and our reason is not correct. So fourth option is the right answer for this question. Now moving to the next question. Next we have is our consider the following plots of an orbital wave function against the distance from the nucleus, the figure representing the two radial nodes in this particular orbital. Now first of all, we know what is a node. A node represents that point where probability of finding electron is 0. Now radial node means that two particular points where your orbital wave function will cuts your your nuclear axis is there. This is your r is the distance of the nucleus. So simply we need to say here, in this particular case your orbital wave function is not 0 because in any particular r value it is not 0. If you see the third option, these two particular points are there where its value is 0 representing the two nodes. So that means which graph is representing the two nodes? The right answer is your option C. And in the option, that is our option 4, which representing the C graph is that particular graph representing two radial nodes. Radial nodes are there. Now moving to the next particular question. Next question says, in an acidic medium, 10 milliliter of a 0.25 molar oxalic acid is titrated with KMnO4. The volume of KMnO4 solution required to reach end point is 10 ml. The strength of KMnO4 solution is. We need to calculate the molarity. So first of all, we know what is our oxalic acid is there. Oxalic acid, this is our oxalic acid. And simply we can write in potassium permanganate KMnO4 is given. Now simply if we write the titration, KMnO4 in acidic medium changes to Mn2 plus, where the oxidation number from plus 7 changes to 2. The N factor here is our 5. Now in this particular case, oxalic acid, which is our C2O4 2 minus, it gives us our carbon dioxide, and if we balance our carbon, we multiply by 2. So in this particular case, if you calculate its oxidation number, that is equals to our plus 3, and in this particular case it is our plus 4. For 2 moles means the change is from plus 6 to plus 8. So here the change is our 2. In order to calculate the strength, simply we can calculate the milli equivalents of oxalic acid is equals to the milli equivalents of KMnO4. Now, milli equivalents, how to calculate? M1, V1 and N1, that is equals to M2, V2 and N2. Now, for oxalic acid, the molarity they have given in the question is a 0.25 and volume in the question they have given is a 10, and then factor we have calculated, that is a 2. And in case of KMnO4, volume, the end point reaches 10 ml. Molarity we need to calculate and its n factor is a 5. So after solving we will get the 0.50 divided by 5 is the strength of our KMnO4, that is a 0.10. So right answer is our second option, which is a 0.10. Now moving to the next question. Okay, I hope you all understand our physical chemistry question. Now Jenny sir will discuss the inorganic question.
As we are done with the organic and physical chemistry, we can say that let's have some fun with the inorganic one, because that was the easiest one out of all the three, right? So let's move to the first question. Chapter wise we are going to solve it. The first question, if we see, I have picked the easier one here. In this, they are asking the highest occupied molecular orbital for any two molecule. Now we know that the atomic number of Ne is going to be 10, and for Ne2 we are having total of 20 electrons to arrange. In this, we are going to start it from 1s2, sigma 1s2, and we know that we are going to end it to sigma star 2pz2, because we have to arrange those 20 electrons. Now for this, we know that the last two electrons are for sigma star. Now let's find the correct option out of all the four options. The correct answer is going to be sigma star P. The second question from the same chapter, match the species from list 1 with their geometry from the list 2. If you look at this, let's find the easiest one for us to address, right? BF4 minus 1. For that, we know that it's a weaker ligand, and because of that, it is going to have AB4 type of construction, and that's why the geometry is going to be tetrahedral. So C is connecting to 1. We are having that for almost 3 options. Let's go for the second one just like that. Again we are having AB4 over here, but now we are having stronger ligand over here. So this time it is going to give us square planar structure. So D is going to connect with the second option. So here we have D with the second option. Here we have D with the second option. Now let's address PCl5. PCl5, we know that it is AB5 type of structure, and for AB5 type of structure, again we are going to have the trigonal bipyramidal structure. For that, these three are the correct answers. Now, out of that, if we see, this is eliminated, this is eliminated. So, we are left with the B1, BRF5, that is AB5, but the bromine is going to have a one lone pair of electron, so it's going to change to square pyramidal. So our correct answer is going to be the third one. The formula for tetraamine aqua chlorido cobalt 3. So alphabetical order we have to follow. Tetraamine, that's why I am going to have NH3, that is tetra, so 4 times. Aqua, so I am going to have H2O. Chlorido, that means I am going to have Cl also, and these all ligands are connected to the cobalt plus 3. Now we know that ammonia is having 0 charge, we know that H2O is having 0 charge, it's neutral, but Cl is going to have a minus 1 charge. So cobalt being plus 3 and Cl being minus 1, the total charge of our cation is going to be plus 2. And to balance that, how many chlorides are going to need? That is the important part for this question. We need 2 of the chloride. If we find our answer, option 1 is going to be our correct answer for this. Here we have two different sentences, two different statements. First one, if we see over here,
They have taken 3-time oxalate with -3 charge, and they are saying that it is an optically active compound as it have chirality in it. Statement 2 is a trans form of this particular compound is correct. Let's address both of them, which one is correct, both of them are correct or not. If you see the first one, if I draw it, you can see that it is going to have a structure of this type. Now, for this one, we know that there is no plane of symmetry, there is no center of symmetry. Both of the points which is making the compound optically inactive is not available over there. That's why we can say that this compound is optically active. Whereas the other one, you can see that both the oxalates are going to be like this, but the H2O is going to be on an axial position, which is going to create a plane of symmetry over there, and because of that it cannot be an optically active compound. So second statement becomes the false one. Now statement 1 is correct and statement 2 is incorrect. That means option 4 becomes our correct answer. Complex which has facial and meridional isomerism. We know that there was a trick where if monodentate ligands we have three of them of one kind and the three of them of the other kind, then this kind of formula can give us both the types of isomerism. Let's check our options. Over here we have both of them as a 2, 2. It cannot show us that. Here we are having that option. So second option becomes our correct answer. This is the easiest question of complete paper for the chemistry actually. 1 by 3 electrolyte in the aqueous solution for the given options. Now, for that, the 1 represents the cations and 3 represents the anions. If the same kind of ratio is followed along with your options, you have your answers. Check once. Here if you see, this compound does not have any cation or anion. That means the given ratio does not apply over there, so that is not our answer. Option number 2. Since Cl is going to be -1, our complex ion is going to be +1, and if we take the ratio, it is going to be 1 by 1. So that is also not our answer. Here Cl is -1 and we are having two of them. So we have double of the anions, whereas the +2 charge will be there for our complex ion. So only one, which is giving us 1 by 2 ratio. So that is also not our answer. So ultimately leads to 3 chlorines, which leads to 3 of the anion and +3 charge on our cation. So it becomes 1 by 3. So fourth option is our correct answer. According to the crystal field theory, the correct order of ligands with respect to their decreasing order of field strength. Decreasing order they are asking, that means I need the highest one first. So, uh, of course, I cannot start with the Cl, so first one is not our answer. Then we have 2 with the carbon monoxide, so either second option or third option is my answer. Look at the second ligand. Here we have ammonia, whereas here we have H2O. That automatically gives us the idea that the order is correct for the second one. So second option becomes our correct answer over here. Among the species given below, the spin only magnetic moment is highest for the given coordinate compounds. Let's check here. So if I see the first one here, in the first one, the titanium is going to be plus 3 and the atomic number of titanium is 22. If it is plus 3, we need to minus 3 electron as well as we are going to minus the 18. That means we are left with only the T electron which is present over there in the 3D. So if we do that, we have only one electron in the D orbital. So here I can write that it has only one electron in 3D. Same procedure if I follow for the Mn, then the atomic number of Mn is 25. Let's calculate the charge of Mn over there. So Mn is having the ion charge minus 3 and one cyanide is going to be having minus 1. So X minus 6 is going to be equal to minus 3, which leads to X being plus 3. Again, we are having the same charge over here. So 25 minus the charge as well as the electrons of last atomic noble gas. Again, 25 minus 21, it is going to lead to 4 electrons. But here we are having the strong ligand which is going to create the shuffling of electrons which leads to ultimately only 2 electrons being unpaired, whereas over here only 1 electron is unpaired. If I follow the same procedure for the third option, atomic number is 26, the charge again 6 cyanide and minus 3, it's kind of a same situation, so it is also going to be plus 3 only. So the amount of electron left over here is going to be 5. Again cyanide is there, reshuffling will occur. So only 1 electron will be remaining unpaired. Now here if I do the same thing, I am going to get 6 electrons as cobalt is having the plus 3 charge. So 27 minus 3 plus 18, which is going to lead to the 6 electron, and ammonia is also going to create the same effect and it is going to rearrange the electron in d orbitals, which leads to 0 electrons. That means wherever I am having the highest amount of unpaired electron is going to give me the highest amount of magnetic moment. That leads to the option number 2. So option number 2 is going to be the correct answer for me here. Among the following options, the correct trend in the electron gain enthalpy. Now, each and every single time teachers have told you that there is one exceptional case where period number 2 is going to have higher amount of negativity, but period number 3 will always have higher amount of negative electron gain enthalpy. So that's why Cl will be the highest one. That's why Cl, F, Br and I, because of the electron-electron repulsions. We know about that. Now, for these two statements, one is labeled as the assertion and another one is going to be the reason. The first ionization enthalpy of oxygen is lower than that of the nitrogen and fluorine. Now we know that the nitrogen is having the half-field stability of p orbital. That's why it is going to have more amount of first ionization enthalpy, whereas oxygen is going to be having lesser amount of it since it is having more electron than nitrogen, so half-field stability is not there obviously. So the order is going to be oxygen followed down by nitrogen and then last comes fluorine. Now in this case, if you read the sentence completely, lower than nitrogen as well as fluorine. So the order is the same thing. So given statement is correct. Now let's find out the reason. When the loss of electron occurs in oxygen, it gets the stability of half-filled p orbital. Of course, the oxygen is having 2p4 electron. When you lose that one electron, it is going to give me 2p3, the half-filled stability, which is why
oxygen is having lower than nitrogen, the first ionization enthalpy. That's why I can say that the reason is also correct and it is explaining your statement. That's why A and R both are correct and R is the correct explanation of A. So second option becomes our correct answer over here. Next one, correct decreasing order of oxidation states. Underline, I'm going to underline it for you because here it is not given. So these are the atoms they want us to give the oxidation state, but remember they want it in the decreasing order. Means the highest oxidation number should be the first one and the lowest oxidation number is going to be the last one. If you see over here for P4O6, I am having 4 of the X where 6 of the oxygen. So oxygen is going to be the -2. The total charge is going to be the 0. So 4x minus 12 is going to be 0, which leads to x being plus 3. So here phosphorus is plus 3. If I apply same kind of logic for the second one, I'm going to get chlorine as a plus 7, which proves that it is not a decreasing order. That means first option cannot be our answer. Look at the second option. In the second option, P4O10. Now, if I do the same thing once again, then instead of 6, I am going to have 10 of the oxygen now. Here 4x minus 20 is going to be 0. So x is ultimately going to be plus 5. So phosphorus is going to be plus 5. We know that in SO3 sulfur is going to be plus 6 and oxygen in water is minus 2. If you see the decreasing order is not being followed. So second option is also not correct answer. In PbO2, Pb is plus 4. In N2O3, nitrogen is plus 3. We know that. And SO3, sulfur is plus 6. Again, it is not followed for these two. So that is also not our correct answer. Automatically, let's confirm the third option as our correct answer. N2O5 nitrogen being plus 5, Al2O3 aluminium, we know that always plus 3, and H2S sulfur being minus 2. The decreasing order is correctly followed. So third option is our correct answer. Let's move to the next question. The lanthanide ions having 4 unpaired electrons. They want 4 unpaired electrons in the given ions. Now we know that the atomic number might be a trouble, so they have given it to us. Right? Look at the first one. For this one, the atomic number is 67. Luckily, for all of the options, they have taken all the ion as a plus 3. That means if the atomic number you take minus the 3 electron which is in the ionic form, at the same time, the same trick, the last noble gas, which is going to be 54, which ultimately leads to the electron of 4f. If you count that, 67 minus 57, which is going to lead and give us 10 electron in the 4f. So here I am having configuration of 4f10. If I follow the same logic over here, so for neodymium, I am having the atomic number of 60, the charge of 3 electron, that's why plus 3, and the last noble gas 54. Again 60 minus 57 going to lead 4f3. So here I am having 3 electron in f orbital. If I do the same, 58 minus 57 will be for the cerium. It is going to give me 1 electron in 4f, and for terbium 65 minus 57, which is going to give me 8 electrons, so 4f8. Now we know that there are total 7 orbitals in the f orbit. If I draw those 7, only the last 4 is going to have 1 electron, whereas the other 2 are going to have paired. That means this is going to be our correct answer because they ask for 4 unpaired electron. The green paramagnetic species, that is a property. By heating KMnO4, it's a standard characteristic of KMnO4 given in the p block elements. So if I have taken KMnO4, if I heat it at given temperature, it is going to give us K2MnO4, MnO2 and O2. And to balance this equation, we know that we have to take only 2 moles of KMnO4. It is going to balance it. And in this, it is clearly written that it's green in color. That means K2MnO4 is going to be our correct answer. Again, we are having 2 statements over here. Generally, 3D transitional metals have high melting points. I am absolutely agree with this because those electrons create more stronger metallic bonds. We know that. At the same time, the 4s electrons are going to create that stronger bonds over there because it is having more less distance with the nucleus as well as more negativity over there, right? So, involvement of 3D electrons, in addition, 4s electron in the interatomic metallic bonding. So, I can say that the reason is also explaining the statement correctly over there. So, both of them are correct as well as giving the right explanation over there. So, A and R both are correct and R is the correct explanation of A. So, second option is our correct answer. Again, 2 statements we have. Heating NaCl with concentrated H2SO4 and MnO2 is going to give us the oxidation of Mn. I don't agree with this. Why? Because in the preparation of halogens, p block elements, we have learned that if any of the halogens you take, in statement 2 also they have taken the same thing only. Any of the halogen if you take with concentrated H2SO4 and MnO2, I am not going to write the balance equation, I am just giving you the heads up. It is going to give us MnSO4, it is going to give us our halogen in the released form. So here we are having -1 oxidation charge, whereas over here we are having 0, which leads to the oxidation, whereas Mn is going from +4 to +2, which is going to decrease the oxidation number. It gives us the reduction reaction. That means it is not the oxidation, it's completely reverse of it. So statement 1 is incorrect. And in the statement 2, if we see they have used the correct word, so statement 2 is correct one. Let's follow the options. Statement 1 is incorrect, whereas statement 2 is correct. So option number 1 is going to be our correct answer. In potash alum, the ratio of potassium ion and the sulfate ion. Before that, we should remember the constitution of potash alum, which is potassium sulfate with aluminium sulfate to crystallize 24 molecules of water. If you simplify this, you are having one of the potassium, you are having one of the aluminium, and total out of 4 you are dividing it by 2, of course, so 2 of the sulfate and 24 divided by 2, 12 of the water. In this, for 1 potassium ion you are having 2 of the sulfates, that leads to 1/2 ratio for the potassium and sulfate ions. The correct statement is, out of these 4, aluminium have 5 valence orbitals. Actually, I do not agree with it because it has the empty 3d orbital also, which automatically leads to total of 9 valence orbitals for aluminium. So, statement 1 is incorrect. Boron has maximum covalency of 4. Of course, boron comes to the second period. It does not have d orbital, which automatically proves this one is the correct one. Let's take the other two for the sake. Beryllium is having 3 valence orbital. Again, beryllium is going to be the same as the boron, and it is also going to have 4 valence orbitals. So the third statement is also going to become wrong. And magnesium has a maximum covalency of 4, which is wrong, by the way. Magnesium belongs to the third period, which leads to the inclusion of 3D. So it can of course have more than 4. So that is also wrong. So second option becomes our correct answer, which concludes the chemistry over here. Now let's have a quick analysis of the complete chemistry paper. I have the organic chemistry in front of me over here, and the questions are total 14. Out of 45 questions, 14 questions were asked from the organic chemistry, and out of that, 1, 2, 3, 4, 5. 5 questions were asked from the grade 11, whereas the remaining 9 questions were asked from the grade 12. In that also, if you see the focus area, aldehyde, ketones and carboxylic acid, they ask 3 questions. Amines, they ask again 3 questions. Biomolecules, they ask 3 questions. General organic chemistry was with the 2 questions, and the hydrocarbon was again with the 3 questions. So there was a selective chapters with a very heavy weightage over there with multiple questions. And what was the level of questions in organic chemistry? See, you can by yourself you can say that I have given a green color to the easier question, I have given yellow color to the moderate question, and the red color for the hard question. Following those colors only, you can see that we are seeing more amount of yellow and the green over there, and that leads to that paper is going to be was actually easy to moderate for the organic part. Now, if we talk about the inorganic one, surprisingly, 6 questions were asked from the coordination compounds. 6 questions out of 45 from a single chapter. It gives us the idea how much importance that chapter carried, and I am sure that teachers has also conveyed the same to you. And again, multiple questions were there from the D block also. 2 questions were from the chemical bonding, and 3 of them were from the combined 11 and 12 chemistry, P block elements. Total of 17 questions were asked from the inorganic chemistry, and if you see and follow the colors over there, you are seeing more of the green over here, which means the paper was again easy to moderate for the inorganic one. For the physical chemistry, here paper was a bit time consuming because of the lengthy calculations, we can say that, and most of the calculations, it's not that we can't do, we can do it, we have done it before, but it takes some time. So I would say that it was time consuming, same number of questions as organic chemistry. Again, if you see here, most of the color you are seeing is yellow color, and that complete part of physical chemistry was divided among all the chapters we have over there, which leads to being it moderate to hard for the physical chemistry. So ultimately, I would say for the conclusion of the chemistry that the paper of chemistry was little bit time consuming, but it was moderate. Thank you.
Okay, so hello friends. Once again, I am back. So now we will do the analysis of physics. That is the last part. So as we discussed during the paper discussion also, that okay, physics paper was shifted towards the moderate, right? But lots of students, like they said, you might have seen some videos, some reels, like some YouTuber bloggers. They are just taking the interview of some student when they are coming out of the examination hall. That how was the paper and all. So they are saying, don't ask about the physics and all. It was very tough. Actually, physics was not that much tough. Okay.
Physics is definitely shifted towards the moderate side, okay. But if you'll see, only few questions were ranked as a tough, hardly one, two or three questions we can say. Otherwise, most of the questions were moderate or easy only, directly formula based or two formulas or we have to use and we have to divide, add, subtract and we'll have to get the answer. Anyhow, let's see. So here, first thing, from class 12, if you will add, then total 12, class 12 over here, okay, okay, sorry, okay. So from here, you can see that class 12, these many questions were there, again these many, and again this, 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 and this. If we'll add all of them, total 24 questions are there from class 12. While from class 11th, obviously 21 questions are there. So we can say that okay, they are nearly at a ratio of 60 to the 40. So ratio was nearly same as they ask always like okay, we always expect that from class 12 few more questions will be there. Generally 60 to 40 ratios they are giving. Now coming to the type of the numericals and all. So here dual nature. So dual nature, modern physics, they are targeting more. Not only here, like over here you can see, then again semiconductors. So like this, here three questions, then semiconductor two questions. So like this five, six questions. So differently they are giving as usual from yeah, whole modern physics. Now over here, de Broglie wavelength, photoelectric effect, all the questions were moderate level. So these questions were moderate because some calculations were there. Apart from that, no hi-fi concepts were required. Only the concept of threshold frequency, wavelength and all was required. Now, second, moving charge and magnetism. Okay, so magnetism they asked one question from solenoid. This was a topic from where generally they don't ask much question. So solenoid they asked one question, and this is a normal like okay field due to like a magnetic field due to a wire like two wires were there, they were attracting, so balancing of force with mg. So generally they used to ask magnetic field due to wire, but this time like different types of wire they will give an structure, but this time they gave something else. They just gave a wire and they use the balancing of force with mg and all. So that's why it got shifted towards the moderate. Otherwise we have seen easy questions on this topic. Now electrostatics. So electrostatics, four questions they asked, among which all of them were moderate, one was easy. This question was good, that is conductors. In conductors, generally they don't ask conductors question directly like this, but here it was asked. Apart from that, capacitor, energy conservation and all, it was a normal question, electric potential energy, normal question. Now, electromagnetic induction. Obviously, we are habituated with this. No surprise was there on this topic. We knew that okay, they will be asking a question and they asked it. Okay. Now, fluids. Fluids, Bernoulli's theorem and terminal velocity. They asked questions from this, which they generally ask. The questions were moderate, but one question on terminal velocity is was tough. Okay, so it was tough. Like at the time of solving, a student faced lots of issues like formulas and all. They are not related with that one. One more question they asked from the fluid, but we kept in SHM because simple harmonic motion. In that they mixed that thing. We will discuss that also. So actually, you can say that okay, they mixed three questions from here. So fluid, they are moving towards it. Every year we are seeing that okay, fluid is becoming the favorite topic or favorite chapter of NEET. Now rotational motion, as usual, like as expected, these topics were there. So no surprises, angular momentum, linear momentum, and moment of inertia. Moment of inertia, normal question was there. So this was simple thing. Thermal properties of matter, volume expansion, easy. Semiconductor again, Zener diode, always expected. We have solved these questions before also. So that's why we kept it in the easy. Depletion width, it is also easy. We have solved these questions before lots of time in PYQ. That's why it was kept like this. Now coming to electricity, current electricity, series and parallel. So no surprises. You can say that it is easy, but we kept it moderate because of its toughness level. Otherwise, it was not very tough. Okay. Gravitation, escape velocity and Kepler's law. Kepler's law directly formula based, escape velocity directly formula based. So there was no surprise from here. Kepler's law is the favorite part of NEET. Like they ask gravitation question, but among that also they have asked lots of time Kepler's law questions. Now alternating current, moderate, it becomes moderate for us also because we are, as being a NEET student, generally we don't like alternating current because sine and cosine equations are there and all. So that's why, so questions was moderate because from that only it is derived. Now thermodynamics, they asked three questions here. Okay, one is polyatomic process, adiabatic process and thermodynamics. So but this was easy. This is also easy. Polyatomic, obviously we are not very much related with polyatomic, that's why it is shifted towards the moderate side. Now motion in a straight line, so moderate, question of average speed and all. Okay, it was not tough. Even we can say that okay, easy, shifted towards the moderate side. Okay, now EM waves, applications and wave equation. So that is a easy only. Directly we have seen that question in PYQ. Then KTG, specific capacity and mean free path. Now this mean free path was also easy, but we are keeping it in moderate because we don't remember the formula, because we use it very less time. Otherwise, this was not tough, not moderate. It was easy only, formula and then ratio, but formula very few students remember it. Now next is like ray optics, apparent depth, normal formula based, lenses, combination of lens, but that's why it was in moderate because two lenses they gave and all. Laws of motion, banking of a road, all of us are habituated to this thing. Atomic structure, bold atomic model, alpha scattering. Now this was easy, but you can say that okay, it was tough also because that graph we don't remember, but we kept it in easy because if you are going through NCRT, as it is our duty to go through the NCRT of all the books in all the subjects for NEET. So that's why I hope all the students remember this graph. So that's why we kept it in easy. Otherwise, it becomes tough if you have not seen that because you can't solve it at the time of exam. Now SHM. SHM, time period related, spring mass system related. Now this is time period related was moderate, spring mass system related was tough. Time period related was moderate because in that one they used fluid and all. That's why I said that okay, fluid, it is the fourth question also you can say. And one more third question, and one more thing, this spring mass system, it was very hard because of the equation of circle. Okay, in this one they gave v versus x curve and then they used time graph, so that's why we kept it in hard level, not a moderate level, because equation of circle is something which we are not very familiar as a student of biology or NEET aspirant. That's why. Otherwise, it was not very tough if you remember the like how to convert ellipse equation into circle equation. Okay. All right. Now unit and dimension. Obviously, dimensional analysis. So it was tough also, guys. Okay, as it is a small topic, simple topic, that's why like okay, we have done this thing before also, that's why we have kept it moderate. But calculation will take time. That calculation was like Boltzmann constant, Stephens constant, all those tough dimensions they gave. But anyhow, this was not tough for us because as a NEET aspirant, we always remember these dimensions. If we are not remembering, then we keep on doing those dimensions in any module, in any notes you will find those dimensions. Okay, anyhow, nuclear physics, q value, easy only, but it was just calculative. Okay, the question was very easy, but it was just calculative. Just we know that okay, binding energy they can't do anything, we have to calculate, so it was calculative. Sound, auger, pipe, not nothing to think about over there, just a like a frequency formula for closed organ pipe, open organ pipe, divide it, you will get the answer. So it was easy only. Now experimental physics, this was something which was tough also for us, because over there they mixed all the experimental theory and we don't remember that. Okay, anyhow we have marked it as a moderate, but we can say that okay, it was tough if we don't remember that what is the like a end correction or index experiment, index correction, all these topic if we don't know, then obviously it will become tough. And as they are not numerical, so during the practice also we don't practice those words again and again, because we have to memorize it only. So that's why you might have missed this question. Apart from that, all the questions were easy only, like lots of questions were easy, as we were expecting that okay, as it is a re-NEET, so it will be a very tough question, as lots of people were saying. But we in the beginning only we said that it will be shifted towards the moderate, not completely tough. But it was just a guess, either we are right or they were right. That is not the question. The question is that how many questions you have done correct. So I hope that okay, you will be getting a good marks in this one. Even if you are getting average marks, then also hope for the best result, because paper was not very easy at that time when you were attempting the question. Definitely a student would have got afraid by seeing this question paper at that